Hey everyone, and welcome to Miss Estruch Biology and this entire AQA A-Level Paper 2 video in which I'm covering all of the theory for topics 5, 6, 7 and 8. And not only that, if you click the link in the description, you can download my workbooks completely for free, which you can complete as you're watching this video to get your own set of notes. However, if you are pushed for time and you're watching this very close to your exams, or you just want to be more efficient with your time, then number one, don't forget you can watch this at ×2 speed. And number two, I've got a ready-made set of notes which cover all of the theory, key terms, key marking points, examiners' tips, summaries, recall questions at the end of each topic, and essay links. Basically everything you need to cover the theory, which is also linked below. But that is it for now. Best of luck with your paper 2, everyone. And we begin with 3.5.1 photosynthesis, and everything in topic 5 is A-level only, not for AS. Now the first part of photosynthesis is a little recap on the structure of a chloroplast, because this is where photosynthesis happens. You have a double membrane organelle, and then the thylakoids are folded membranes which contain photosynthetic pigments such as chlorophyll, and these are going to be involved in the light-dependent reactions in photosynthesis. You also have this fluid-filled centre called the stroma, and that is where the light-independent reactions happen. Now, chlorophyll is actually made up of many photosynthetic pigments, and these are in those proteins embedded within the thylakoid membrane. And the advantage of there being multiple photosynthetic pigments is each of those pigments absorbs light at a different wavelength. And that then means there's a bigger spectrum of visible light that is able to be absorbed by the plant. And if a bigger spectrum of wavelengths can be absorbed, that means overall more light energy is absorbed. And if more light energy is absorbed, then there's more energy for the light-dependent reactions. So the light-dependent reactions then, this is the first stage in photosynthesis, and it happens on the thylakoid membranes, or a stack of them is known as a granum. The light-dependent reactions require light energy and they use water, and they produce ATP and reduced NADP. And that is through these three stages: photolysis of water, photoionization of chlorophyll, and chemiosmosis. So we're going to go through those three and see how it results in the production of ATP and reduced NADP, which are needed for the light-independent reactions. Photolysis of water, photo, if you see that in biology, it means light, lysis means splitting. So it is using light energy to split water. That is what photolysis is. And we've got an equation here. Water, and it's using the light energy, is split into oxygen, electrons and protons, also known as hydrogen ions. The protons are going to be used to reduce NADP, and that helps to maintain a proton gradient in one of the later stages. Electrons, those are going to replace the electrons that get lost in chlorophyll in one of the stages we look at. Oxygen, this is a byproduct, it's not actually used. So a few things just to point out here. NADP is a coenzyme. These are not enzymes. They don't catalyze reactions. They are going to be carrying molecules from one point of a reaction to be used later. And if it's reduced, it means it's gained a proton and an electron. So that is what we mean by that term. Photoionization of chlorophyll. This is when light energy, again, so photo meaning light energy is absorbed by the chlorophyll, and that energy causes an electron to be released, so it's been ionized. But they are really specific with the terminology you use for this. So you'd have to say the light energy is absorbed by the chlorophyll. That's usually 1 mark. Then it's saying what that light energy does. So it results in the electrons becoming excited and raising up an energy level. That is the language they put in the mark scheme: excited and raising up an energy level. And that causes the electrons to be released, and therefore chlorophyll has been ionized. And those electrons are going to be used in one of the later stages to help make ATP and reduced NADP. And that later stage I keep referring to is chemiosmosis. This is where we're going to see the protons and electrons being used to make ATP and to make reduced NADP. NADPH is the same thing as reduced NADP. either of those will be accepted on the mark scheme. So step 1, here is PS2. You don't need to know that terminology for AQA. That is just a protein embedded within the thylakoid membrane. And we can see here, this is the thylakoid lumen. This is the chloroplast stroma. So within the thylakoid lumen, so within inside of the thylakoids, that's where we have our photolysis happening. Water has been split into protons and electrons, which are picked up by this protein, and the oxygen's a byproduct. So we can see there we've got our electrons, and those are replacing the electrons that are getting excited, raising up an energy level and leaving the chlorophyll, moving on to the next protein in the photoionization of chlorophyll. So we get these electrons moving along this chain of proteins, and every time they move from protein to protein, they release energy. And that energy is used to pump protons from the stroma into the thylakoid lumen. And as a result, from those protons being pumped across and the protons released from photolysis of water, we end up with lots of protons on the thylakoid lumen side compared to the stroma, which means we've created an electrochemical gradient. Those protons can move down that electrochemical gradient back into the stroma, but it has to be through a protein, and the only protein they can move through is the enzyme embedded within the thylakoid membrane, ATP synthase. So the protons move through ATP synthase, and that enables that enzyme to catalyze the phosphorylation of ADP into ATP. So that is how ATP is made in chemiosmosis. The last step then is, how is this reduced NADP made, or NADPH? Well, the protons that have moved through ATP synthase are picked up by that coenzyme NADP, and also the electrons, when they get to the end of this electron transport chain, they are also picked up by NADP. And that then forms reduced NADP, or NADPH. And that's how we get those two products that we're now going to see used in the light-independent reactions, also known as the Calvin cycle. This stage happens in the stroma. It uses the enzyme Ribisco, so it's temperature sensitive. So the key things you need to know about the Calvin cycle are, it's going to use carbon dioxide. It also uses reduced NADP and ATP. The ATP is going to be hydrolyzed to provide energy for reaction. The reduced NADP is going to donate hydrogen, which is a proton and an electron, and therefore it will reduce molecules in the cycle. We're going to have a look at this whole cycle, but that's just a summary to be aware of. So here is the cycle. Step 1 is carbon dioxide is going to enter the cycle, and it combines with RUBP, which is a 5-carbon compound. So a 5-carbon compound, RUBP, is going to combine with carbon dioxide, a 1-carbon compound, and that is catalyzed by Ribisco. So that adds up to 6 carbons, but we actually get that splits to make 2 3-carbon compounds known as GP. So all of that is step number 1. Those 2 molecules of GP are then reduced to form 2 molecules of triose phosphate. So the way that they are reduced is using the reduced NADP. So the hydrogen from reduced NADP is gained by the GP molecules to reduce it. The ATP is hydrolyzed to provide the energy for that reduction. From that triose phosphate, 1/6 of that molecule goes towards making a hexose sugar every turn of the cycle. So that's what we mean by useful organic substances. Anything that contains carbon, for example, it could go on to make glucose, which will be a respiratory substrate for the plant. The other 5/6 of that triose phosphate are used to regenerate RUBP, and we need energy from ATP for regenerating that RUBP. Now, those hexose sugars or organic substances that are made, whilst glucose is the main product that is made in terms of those hexose sugars, that monosaccharide can join to form disaccharides in the plant, such as sucrose, and polysaccharides such as cellulose to make the plant cell walls, and starch as well as a storage of that glucose. It can also be converted into glycerol and therefore combined with fatty acids to help make amino acids, and it can also be combined with nitrates to make amino acids and therefore proteins in the plant. There are limiting factors that reduce the rate of photosynthesis. Those are the light intensity, carbon dioxide concentration, and temperature. The light intensity is going to limit the rate because it provides the energy in the light dependent reactions. So if you don't have a high enough light intensity, then photolysis of water and photoionization of chlorophyll won't be happening at a fast rate. However, it will get to the point where it can get brighter and brighter and the rate still remains the same because maybe you now have a different limiting factor such as carbon dioxide concentration or temperature. Carbon dioxide concentration can limit the rate because it is the reactant in the Calvin cycle. Again, it will plateau eventually because maybe you don't have a high enough light intensity or the optimum temperature. Temperature is linked to the idea of it being an enzyme-controlled reaction. So at too low a temperature, there's not enough kinetic energy for successful collisions, and therefore you don't get as many enzyme-substrate complexes. And too high a temperature, the enzyme denatures, active sites change shape, so again you don't get as many enzyme-substrate complexes. Now, for maximum photosynthesis, and therefore plant growth, in farming and agricultural practices, they want to be removing limiting factors so they get more crop to sell. And that could be by increasing the light intensity, the temperature, and burning fuel to release more carbon dioxide. But all of those strategies cost money. So they will have to work out if the extra growth from photosynthesis offsets the cost of paying for extra heating, lighting and fuel. And this often comes up as application questions linked to experiments where you have to work that out. Next is 3.5.2, respiration. There's aerobic and anaerobic respiration, and we are going to start with aerobic, which is split into 4 key stages. We have glycolysis, which happens in the cytoplasm of the cell, but the other 3 stages do happen in the mitochondria. The link reaction and the Krebs cycle happen in the mitochondrial matrix, which is the fluid-filled center. And oxidative phosphorylation happens on the cristae, or the inner mitochondrial membrane. And this is an enzyme-controlled reaction, so it is sensitive to temperature changes and pH changes. The respiratory substrate typically is glucose, and that is broken down through the reactions and it will release ATP. But if there's no glucose, then glycogen in animals could be hydrolyzed into glucose, or if it's a plant, then the starch could be hydrolyzed into glucose. Now, if there's no glucose or glycogen in animals or starch in plants, then the next option is other biological molecules will need to be used. So lipids can be hydrolyzed into glycerol and fatty acids. The glycerol then gets phosphorylated into triose phosphate, and it can enter aerobic respiration at the Krebs cycle to be used. Proteins can also be modified and used. They'd have to be hydrolyzed into amino acids, and then the amino acids would have to be hydrolyzed and the amine group removed, or deamination. The carbon compound that remains could then enter the Krebs cycle. So let's have a look at all of these stages, starting with glycolysis, because that's the first stage. And this actually happens in both aerobic and anaerobic respiration. No oxygen is needed in this stage, which is why it's in both types of respiration, and it happens in the cytoplasm. So there's 3 key stages in glycolysis. Phosphorylating glucose to form glucose phosphate, and that requires ATP. The production of triose phosphate and the oxidation of triose phosphate to produce pyruvate with the net gain of ATP and reduced NAD. So NAD is one of the coenzymes used in respiration, and it has the same role as NADP did in photosynthesis. Do not confuse those though, it will cost marks. So the way that I always say to remember it is NADP, think P for photosynthesis. So here is the glycolysis reactions. We start with glucose, a 6-carbon compound, and we said that is phosphorylated to glucose phosphate. phosphorylation means adding on phosphate groups. And that's why we have to use ATP. 2 molecules of ATP get hydrolyzed, and that means the inorganic phosphate removed from those 2 molecules can be added onto glucose, and that makes glucose phosphate. That is a more reactive molecule, and that causes it to split into 2 molecules of triose phosphate. That triose phosphate is then oxidized to make 2 molecules of pyruvate. And we can tell it must be an oxidation because the coenzyme NAD has gained a hydrogen from the triose phosphate. And if it's gained a hydrogen, that means it's a proton and an electron. So if triose phosphate has lost that proton and lost the electron, then it has been oxidized. And the NAD has been reduced because it gained that electron in the hydrogen. So you can call that NADH or reduced NAD. Now this oxidation reaction also produces 2 molecules of ATP. So this means from 1 glucose molecule, we get 2 pyruvates, 2 reduced NAD, and a net gain of 2 ATP molecules. And that's because although we've made 4 in total, we had to use 2 to start with. So it's a net gain of 2. Mark schemes are incredibly picky that you say net gain of ATP. Even better if you can say a net gain of 2. So now we've got our pyruvate and those 2 molecules of reduced NAD. Those are going to be actively transported into the mitochondrial matrix to be used in the next stages. The ATP can be used in the cytoplasm for other reactions. The link reaction is the next step. And this is when pyruvate is going to be oxidized further into acetate. And again, we can tell it's an oxidation reaction because NAD has now formed reduced NAD. So NAD has been reduced, which means pyruvate must have been oxidized. 1 carbon has also been removed, so we've formed 1 carbon dioxide molecule. So that means we've gone from pyruvate, which is 3 carbons, to acetate, which is 2 carbons. Now, acetate can't enter the Krebs cycle as it is, so it has to combine with a coenzyme called coenzyme A to make acetyl-CoA to enter the Krebs cycle. So that's the link reaction. And for every glucose molecule, we end up with 2 acetyl-CoA, 2 carbon dioxide, and 2 reduced NAD. And the reason for that is, for every glucose molecule, 2 pyruvate molecules were made in glycolysis. The Krebs cycle then, and this is actually just showing you the link reaction leading into the Krebs cycle. Acetyl-CoA is going to combine with a 4 carbon molecule. So we've got 4 plus 2, that makes a 6 carbon molecule. And once that has happened, coenzyme A is released and it can be reused in the link reaction. We then have a series of redox reactions where we go from having a 6 carbon molecule back to the 4 carbon molecule. And you don't need to know all of the intermediates. You just need to know that when you go from that 6 carbon molecule back to the 4, there's a series of redox reactions. And the carbon compounds are being oxidized. The coenzyme NAD and FAD are being reduced. And we get 3 reduced NAD being made and 1 reduced FAD. Because we're going from a 6 carbon to a 4 carbon, we've also lost 2 carbons to create 2 molecules of carbon dioxide. And that whole stage also releases 1 molecule of ATP. So the product per cycle is 3 reduced NAD, 1 reduced FAD, 1 ATP and 2 carbon dioxides, but per glucose molecule, which is really how we should do it, because in respiration, in aerobic respiration, we will have 1 glucose molecule entering, which means the cycle will happen twice because we get 2 pyruvate, therefore 2 acetyl-CoA. So that means from 1 glucose, we have 6 reduced NAD, 2 reduced FAD, 2 ATP and 4 carbon dioxide molecules. So if we have a look at everything we've made so far then, we have got in total from glycolysis, link reaction and Krebs cycle, 10 reduced NAD, because we had 6 made here, 2 made here, and 2 made here, taking into account how many times each of these processes occurs. We have 2 reduced FAD from the 2 lots of the Krebs cycle. We have a total of 4 ATP molecules so far. We had a net gain of 2 in glycolysis, and we had 2 from the Krebs cycle happening twice. And we have 6 carbon dioxide, which is waste, it's not useful, but we do have 6 carbon dioxide molecules. We had 2 being produced in the 2 lots of the link reaction, and 4 being produced in the 2 lots of the Krebs cycle. These types of questions are becoming more common, where you have to say how many of a particular molecule you have and identifying where it came from. So you do need to make sure you know all of this chemistry. So the final stage is oxidative phosphorylation. And this is when most of the ATP is made, because so far we've only made 4, a net gain of 4, which is not enough for metabolism. And this stage involves the transfer of electrons and the movement of those electrons along that inner mitochondrial membrane and the protons moving across. So in this one, first of all of those reduced coenzymes that we made are going to accumulate here in the mitochondrial matrix. The hydrogen is removed, and that is then split into protons and electrons. The electrons are picked up by these proteins and are passed down a series of electron carrier proteins embedded in that inner mitochondrial membrane, and this is the electron transport chain. As they move from protein to protein, they release a small amount of energy, which is used to pump the protons from the mitochondrial matrix into the intermembrane space, which is the gap between the inner membrane of the mitochondria and the outer membrane. That then creates an electrochemical gradient, meaning lots of this ion, the hydrogen ion, on one side. And as a result, the hydrogen ion can move down its concentration gradient through the enzyme ATP synthase. And that enables ATP synthase to phosphorylate ADP, meaning ADP plus PI, into ATP. So that is how we get lots of ATP being produced. And this is almost identical to that chemiosmosis that we saw in photosynthesis as well. Final thing then is the role of oxygen. And this is a really common key marking point. Oxygen is the final electron acceptor. What that means is oxygen will pick up the electrons at the end of the electron transfer chain. And it picks up those electrons. It also picks up the protons, and that then forms water. And it's really important that oxygen does this, because by picking up the electrons at the end of the chain, it means the electron transfer chain can continue, so protons continue to be pumped, and therefore ATP continues to be made. Anaerobic respiration is when there is no oxygen, and it only happens in the cytoplasm. The pyruvate that's produced in glycolysis is reduced instead of oxidized. And it will form ethanol and carbon dioxide if it's a plant or a microbe, or lactate or lactic acid in animals. And this is by the pyruvate being reduced because it gains the hydrogen from reduced NAD. That reoxidizes the reduced NAD into NAD so that it can be used in glycolysis again to make sure that at least glycolysis can still continue even though there's no oxygen, so that you get some ATP. So let's just have a look at that then. Here's glycolysis, as we just learnt, and in animals, lactate is made. So that pyruvate is reduced into lactate by gaining the hydrogen, which is a proton and an electron. That NAD that has been made or regenerated, reoxidized, can then be used in glycolysis again, so at least you still continue to get a net gain of ATP. Exactly the same in plants and microbes, but the product's different, ethanol and carbon dioxide. Next then is 3.5.3, energy and ecosystems. Now in any ecosystem, plants are going to be the producers in a food web. And that's because they produce their own organic compounds from photosynthesis, like we just saw. And between each trophic level, meaning the stage in the food web, energy is lost. And that's because between each stage, the organism will be respiring, so using up that glucose, and therefore there is less biomass available in the organism. And also there'll be energy lost through excretion as well. Everything that remains, that is available for the next organism to consume, is the biomass, and the amount of biomass remaining in the organism can be measured in terms of mass of carbon or dry mass of tissue per given area, but usually it's mass of carbon per given area. So to measure that dry biomass, if you wanted to know what the amount of biomass was, because it is the dry mass or the mass of carbon, you have to dehydrate it. And in order to dry out your sample to measure the biomass, you have to heat it in an oven at temperatures lower than 100 degrees C. Now, this has come up a few times recently, and students didn't get the mark unless they said heat. You can't just say put it in an oven. You have to specify heat it in an oven, and it has to be at a temperature lower than 100 degrees C, because anything above that will cause it to burn, and if it's burning or combusting, you're making carbon dioxide, and that is removing carbon. So heat it in an oven lower than 100 degrees C, and you need to constantly weigh while you're doing this drying, and then when the mass becomes constant, that means you now have just the dry mass remaining because no more water is evaporating, which is why the mass would have been decreasing. The chemical energy stored in dry biomass can also be measured or estimated using calorimetry. And this is done by burning the sample in a bomb calorimeter. And then the energy released from that combustion is used to heat a known volume of water. What you would do is measure the temperature increase, and that is then used to calculate the amount of energy that there must have been in the biomass, because that energy has been converted into heat energy, so it'd be the temperature increase. And that is measured in joules or kilograms per gram. Next is known about NPP and GPP. How productive an ecosystem is depends on the abiotic and biotic factors. So if there's plenty of favourable abiotic factors, water, a high light intensity, optimum temperature, then green plants will be able to photosynthesize at their maximum rate and therefore produce more organic substances such as the carbohydrates, and therefore it's more productive. And this can be quantified using GPP and NPP. GPP, which is gross primary production, that is the chemical energy stored in plant biomass in a given area or volume. So basically it's the total energy resulting from photosynthesis. Net primary production, or NPP, that's the chemical energy stored in plant biomass after respiration losses have been removed. And that is basically what is available for the next organism in the food chain, because you've taken away the losses, the energy losses already. Now, if you are asked what either of those terms mean, always give the equation, because that is always worth a mark. The NPP, as I said, that is the energy left over after respiration has been removed, and therefore it's what there is available to make new biomass. Another equation you need to know is the net production of consumers in a food web. And to work out the net production of consumers, known as N in this formula, you would need to do I, which is the chemical energy stored in the ingested food, minus the energy lost. And animals can lose energy through feces and urine and respiration. So we can see we've got our energy input through the ingested food minus the energy lost through respiration and through feces and urine. The productivity refers to the rate at which biomass is produced. Primary productivity is the rate at which producers convert light energy into chemical energy or biomass via photosynthesis. Secondary productivity is the rate at which the consumers produce biomass by eating other organisms. And the rates of productivity are recorded using these units: kilojoules per hectare per year. So kilojoules is the unit for energy, but we do it per area and per year. And the reason we do it per area is so that we can standardize the results, taking into account that if you have a bigger area, you probably have more plants, so you would have more energy. And we do it per year to standardize the impact of the seasons. Because if you measured one area in summer and another in winter, summer would have a higher productivity because there's more plants photosynthesizing. Next is 3.5.4 nutrient cycles. Nutrients are recycled within natural ecosystems, and we see this in the nitrogen cycle and the phosphorus cycle for AQA. And microorganisms play a vital role in both of these cycles to recycle the elements phosphorus and nitrogen. Mycorrhizae are the fungal associations that you get between plant roots and beneficial fungus. And this is going to be useful because the fungi has these hyphae which can branch through the soil and increase surface area for water and mineral absorption. The mycorrhizae also act a bit like a sponge. They can really absorb and retain water. So that makes it quite drought resistant because that water and minerals is going to be shared with the fungus and the plant. So this is vital in the nutrient cycles because if there is a soil which is quite low in either nitrate ions or phosphate ions, having mycorrhizae present will increase the ability to absorb that for the plant. And this is a mutualistic relationship because the plant also provides a benefit to the fungus because plants photosynthesize and it can share some of those carbohydrates with the fungus. So let's look at the nitrogen cycle. The nitrogen cycle is a really important one because nitrogen is needed for making proteins, ATP and nucleic acids. And the challenge in this cycle is, although nitrogen is very abundant in the atmosphere, nitrogen gas has a triple bond between those two nitrogen atoms, and we do not have an enzyme that can break it. However, microorganisms do. So microorganisms are incredibly important in this cycle. There's 4 key stages: ammonification, nitrification, nitrogen fixation, and denitrification. All of these stages use microorganisms. So step 1 is nitrogen fixing or nitrogen fixation, which I've labelled on the diagram. This is when the nitrogen from the atmosphere is fixed by bacteria, which means bacteria break that triple bond between the N2, the 2 nitrogen atoms, and it fixes it, meaning converting it into a nitrogen-containing compound, which is ammonium ions. Now that can either be done by bacteria that are free living in the soil, so the ammonium ions are fixed into the soil, or there are some plants, such as leguminous plants, which are clover and beans, that have these nodules, which means lumps, on their roots. And within those root nodules, there are bacteria that can fix that nitrogen directly into the plant itself. Step 2 is nitrification. This is a 2-stage oxidation reaction. The first step is those ammonium ions are nitrified into nitrites by nitrifying bacteria. Nitrites are then oxidized further, again by nitrifying bacteria, into nitrates. And those nitrates can then be absorbed by the root hairs of the plant and be assimilated, meaning used in the plant and converted into proteins or DNA. And to get into animals, animals eat the plants. Ammonification links to the death and decomposition of animals and the decomposition of plants. But here is where you have to be really particular with your language to get the mark. You have to name the molecule that would be in the dead animal or plant or in the waste. You can't just say a dead animal or plant is broken down. So for example, proteins in a dead animal or proteins in a dead plant, urea, which is an example of the waste, or DNA in the dead animal or plant, you'd have to name it. Those are then decomposed by saprobionts. And saprobionts are bacteria and fungi that can digest dead matter or waste outside of a cell, so extracellularly, and that then reforms the ammonium ions. Now the last stage I've not actually labelled on, it's over here, nitrification. This is not a useful stage because this is when those nitrates could be converted back into nitrogen gas, and therefore you're losing nitrogen compounds that could be absorbed. This only happens if there are anaerobic conditions, and that would then mean that denitrifying bacteria that do this process would be able to thrive, and these ones that are all aerobic bacteria wouldn't. So this is why it's so important to keep soil aerated, so to plough it and toss the soil before you replant seeds, to make sure that there's plenty of oxygen so that you get the beneficial stages happening and not the denitrification. It's also why having waterlogged soils can be an issue, because the water displaces the air and then you'd have the denitrifying bacteria as well. Final note is a key tip for the exam, and that is making sure that you are saying nitrogen containing compound for any of these stages. We only say nitrogen when it is literally the element in the atmosphere. All of these other stages, we have a nitrogen containing compound. So then we have the phosphorus cycle, and phosphorus is really important because it's used to make the phosphate group in DNA, RNA, ATP, and in a phospholipid to make the phospholipid bilayer. And this cycle is quite different to other ones because there isn't a stage where you have it as a gas in the atmosphere. And instead, you mainly have it stored in phosphate ions in the mineral form in rocks. So here's the phosphorus cycle. If we start from phosphate ions in oceans and soils as a store, it's absorbed into the plant by active transport. That would then be converted into maybe DNA or phospholipid bilayer. Animals would consume that, and that's how it gets into animals. We would have some return to the oceans by decomposition of the plant and decomposition and excretion from animals. But we also have phosphate ions from waste and remains, such as bones and shells that don't get broken down as rapidly. And also guano. Guano is this bird pea, this white bird pea that you can see here, which is very rich in phosphorus. Now, those bones and shells that aren't going to be decomposed by the saprobionts will be broken down through erosion over time, and that returns the phosphate ions back to the ocean and soils. But we can also have rock formation happening. This could be deposition or sedimentation. So those phosphate ions within the water or in the soil, over many, many years, heat and pressure, it creates those phosphorus sedimentary rocks. And to get that back into the water, again, it'd be erosion. Now, fertilizers, this is going to be something that you'd have to artificially add to get more phosphorus back into the soil. So here are our key stages. We've said the weathering of rocks, and that releases phosphate ions into soils and water. Plants absorb phosphate ions through their roots. Animals obtain phosphorus by eating plants or other animals. Excretion and death will return phosphorus to the soil or water. And decomposers, so those saprobionts, can break down dead organisms, releasing phosphate ions into the soil. Fertilizers are really, really important to replace nitrate and phosphate ions lost when either plants or animals are going to be harvested and removed from the cycle. Because if you're removing a plant or an animal from your cycle, then you're removing those mineral ions that are locked up in their cells, and also they're not there to decompose and return it back to the soil. So you always have to use fertilizers, and that can either be natural, so using manure, because we know from those two cycles that in feces there are phosphorus, phosphate ions and nitrogen containing compounds that can be broken down. Or you can use artificial fertilizers, which are inorganic chemicals, meaning no carbon, and there are pros and cons of these too. Natural fertilizers are cheaper. If you have animals on your farm already, it's free, it's just their manure. However, you don't know the exact mineral content, so you don't know how much nitrate or phosphate there is in there, and you can't control that. So artificial fertilizers, that is their main advantage. You can control the exact minerals and the proportions that you have. But you do have to pay for them, so they're going to be more expensive, and they're more harmful for the environment. And that's because inorganic substances are more soluble. Therefore, when it rains, they're going to dissolve in that rainwater, and they can then be washed away to water sources and have a negative impact on the environment. So the sorts of environmental impact, first of all, leaching, and that is when water-soluble compounds, such as the nitrates or phosphates, get washed away from the soil into rivers or ponds or lakes nearby. And if nitrogen fertilizers leach into waterways, that causes eutrophication. So eutrophication is when nitrates have leached from the fertilized field into the pond or lake, whatever water source it might be. And that causes the algae in the lake to grow much more rapidly. That creates a blanket of algae across the surface, and that blocks sunlight from reaching the lower levels in the water and any lower down plants. So if those plants lower down aren't photosynthesizing, they die. Bacteria within the water will then decompose those dead plants, but in order for bacteria to do that, they have to respire more, which means they're going to be using up oxygen. So using up all that oxygen eventually means there's not enough oxygen dissolved in the water for all of the other aquatic organisms, such as fish. And if fish don't have oxygen, they can't respire and they die as well. AQA topic 6, and we begin with 3.6.1 stimuli, and this is looking at the internal and external that can be detected and lead to a response. Starting with your definition of a stimulus. This is a detectable change in the environment, and these stimuli are detected by cells known as receptors. And an organism increases their chance of survival if they can respond to stimuli via different response mechanisms. So for plants, there is response as well. And we begin by looking at tropisms, which is the term given to when a plant responds to a stimulus, and they respond via growth to that stimulus. A positive tropism means that the plant would be leaning towards or growing towards the stimuli. Negative means moving or growing away from it. And the two that you learn about are the stimulus of light and gravity. Tropisms are controlled by specific growth factors, and the one example you learn about is indole acetic acid, or IAA, and you can abbreviate to that in the exam. And IAA is a type of auxin, and therefore it can control cell elongation in the shoots and inhibit the growth of cells in the roots. It's made in the tip of the root and the shoot, but then it diffuses to other cells within the shoot and the root. So if we have a look at phototropisms, first of all, photo meaning light, so this is the response to light. In the shoots, light is needed for the light-dependent reactions that you learn about in topic 5. So the plant is going to benefit by gaining more light energy and therefore leaning or bending towards the light source, the stimulus. And this is positive phototropism. So the shoot tip is where IAA is produced. IAA will diffuse to the shaded side. So if you've got light from one side over here, the IAA will diffuse to the shaded side. And that is what we call unilateral light source, and therefore we get this uneven distribution of IAA. And the cells that are exposed to the IAA are going to elongate more, and as a result, the shaded side cells will be longer than the light side, so it's longer and it bends over towards the light source. In the roots don't photosynthesize. They don't have chloroplasts and chlorophyll, so they don't require light energy. So there's no advantage of growing towards the light source. And instead, there's actually an advantage to grow away, because that means the roots are anchoring the plant deeper into the soil. And in the roots, if cells are exposed to a high concentration of IAA, that inhibits cell elongation. So you still have IAA being produced in the tips of the root, but the IAA diffuses to the lower side, so the direction where you would have gravity, the lower side, and that inhibits the elongation of the cells on the lower side of the root. The ones on the top side are still going to grow, and therefore that top side is longer and it bends downwards away from the light source and towards gravity. But this would be our negative phototropism because it's away from the light source. With regards to gravitropism, in the shoots, again, IAA is produced in the tip of the shoot, and IAA will diffuse from the upper side to the lower side of a shoot. And if a plant is vertical, not lying down like we can see in this picture, this causes the plant cells to elongate and the plant grows upwards because the IAA would be moving down towards the bottom, and those bottom cells will grow more, and it just makes the shoot grow upwards. If a plant is on one side though, like we can see in this picture, the side that is exposed to the auxin will elongate, and that can cause it to grow upwards. And that would be negative gravitropism. It's growing away from the direction of gravity. In the roots, it's the opposite. The IAA will diffuse to the lower side of the root so that the upper side elongates, and the lower side doesn't, so the root bends downwards towards gravity, which is a positive gravitropism. Next then, the other simple responses are taxis and kinesis, which keep organisms within their favourable conditions of their environment, for example, light intensity, moisture levels and chemicals. Taxis, this is when an organism will move its entire body towards a favourable stimulus or away from an unfavourable stimulus. So this is directional. When an organism moves towards a stimulus, this is known as positive taxis, and when it moves away, it's negative taxis. Kinesis then, this one is not directional, but it's a change in the speed of movement and the rate it changes direction. If an organism moves from an area where there are beneficial stimuli to an area where there are more harmful stimuli, its kinesis response will be to increase the rate it changes direction, and therefore it's more likely to locate and return to the favourable conditions again. However, if it tries that and the organism is completely surrounded by negative stimuli, so no matter how many times it changes direction, it can't find favourable conditions, then the rate of turning actually decreases to keep it moving in a relatively straight line to increase the chances of it finding a new location with favourable conditions. And just linked to this, most of the responses will be that they'll want an area where it's dark, and that would be to not be easily seen by predators or to stop them from drying out. Moisture, they'll want a more moist area, again, to stop them from drying out. Chemicals, they might be moving towards certain chemicals because that might indicate the presence of food. Simple reflexes are something that we have in more complex organisms with a nervous system, and this would involve a receptor detecting a stimulus. That would then result in coordination occurring with the coordinator, then a response carried out by an effector. And the nervous system is made up of the peripheral and the central nervous system. The peripheral, abbreviated here to P, nervous system, NS, is the receptors, sensory and motor neurons. The CNS, or the central nervous system, that's the coordination centres, so the brain and the spinal cord. So here we have a simple reflex, and a reflex is a rapid automatic response to a stimulus. Reflexes are involuntary, so they do not require conscious thought. You don't have to learn to respond in that way to protect yourself. And the point of a reflex is it protects against damage to body tissues, and it could also help you to escape from a predator. There are 3 neurons in a reflex arc, so it's very, very simple. You have the sensory neuron, which is going to be connected to the receptor. That detects a stimulus and sends an impulse to the spinal cord. The relay neuron is going to be located in the central nervous system, which will be the spinal cord for a reflex. And then the motor neuron that carries the impulse to an effector to carry out the response. And the response in terms of a reflex reaction is very fast because the impulse bypasses the conscious part of the brain and it travels through the reflex arc. And as we can see, the reflex arc only contains 3 neurons and therefore 2 gaps, 2 synapses. And because we've only got 2 synapses, that means it's faster because it's slower transmission at synapse because it's diffusion of a neurotransmitter. Now, actually, in this image, you can't see the relay neuron, but there would be a sensory neuron, relay neuron here in the spinal cord, and then the motor neuron. Receptors detect a stimuli, as we've said, but there are specific receptors that you need to learn about. And each receptor responds to a different stimulus, and this stimulus of a receptor leads to the establishment of a generator potential, which can then cause a response. And we'll come to the idea of establishing a generator potential and an action potential shortly. So the 3 receptors that you need to know are the Pacinian corpuscle, rod cells and cone cells, both of which are in the retina in the eye. So your Pacinian or Pacinian corpuscle, this receptor responds to pressure changes. They are the receptors that you find lots of deep in the skin, mainly in the fingertips and in feet. And it consists of a single sensory neuron wrapped in layers of lamellae separated by gel. The sensory neuron seen in corpuscle has a special channel protein, or has special channel proteins in its plasma membrane. So here's our structure we can see. And those special channel proteins are stretch-mediated sodium ion channels. And these will open and therefore allow sodium ions to diffuse into the sensory neuron when those channels are stretched and deformed. And when pressure is applied to a fingertip or feet, that is applying pressure to the skin, and these layers of lamellae get pushed and deformed, and that stretches and deforms the shape of the sodium ion channels so that they open and sodium ions diffuse in. So when pressure is applied, it causes these sodium ions to diffuse in, and that will then result in the establishment of a generator potential. More on that very soon. Next, then we look at the rod and cone cells, which are the two photoreceptors, or light receptors in other words, in the human retina. Rod cells process the images in black and white. To create that generator potential, the pigment inside of these rod cells, known as rhodopsin, has to be broken down, and that is done by light energy. They can detect light of very, very low light intensities, as many rod cells connect to one sensory neuron. In this case, we can see it's called a bipolar cell before it gets that sensory neuron. And this is known as retinal convergence because we have multiple rod cells converging or meeting at one bipolar cell. And this means that collectively they are able to trigger that action potential, but because you have got multiple rod cells feeding to one bipolar cell, it means the brain cannot distinguish between the separate sources of where that light energy came from. So that's known as low visual acuity, which means you don't have that sharp, clear, crisp vision. So when it's very, very dark, you can still see, but you only see in black and white, and it's blurry. So next time it's nighttime, have a look and you'll notice you pretty much only see in black and white, and you don't have that clear, crisp vision, meaning low visual acuity, and that is why the retinal convergence. Cone cells process images in colour. There are three types that contain different types of pigment known as iodopsin that you get in the cone cells. You have red iodopsin, green and blue iodopsin, which all absorb different wavelengths of light. Now, iodopsin found in these cone cells is only broken down in high light intensities. So you do need a high light intensity to be able to see in colour vision. You also need to have a high light intensity to see in colour vision because you have one cone cell connecting to one bipolar cell, and therefore you need to have a high enough stimulus to trigger that action potential. So when it is much brighter light intensities, you can see colour, and you have a high visual acuity because you've got one cone cell connected to one bipolar cell, so the brain can distinguish between separate sources of light. Now, the distribution of the rods and cone cells is uneven in the retina of the eye. And here is the fovea. That is where the highest light intensity hits the retina because of where the light is focused through the lens. So at the fovea, you have most cone cells. We can see here that's where the fovea is. You have lots of cone cells there. Whereas the rod cells, because they still work at lower light intensity, those are mainly located further away from the fovea because they'd still work in that location. Next, then we move on to the control of the heart, and cardiac muscle is myogenic, and that means it contracts on its own accord without any input from the nervous system, but the rate of contraction is controlled by the nervous system and also electricity. So that's what we're going to have a look at. The sinal atrial node, or SAN, is located in the right atrium, and that is known as the natural pacemaker. The AVN, or atrioventricular node, is located near the border of the right and left ventricle. It is still within the atrium, though, just here. You have a bundle of His which runs through the septum, and then the Purkinje fibres branch into the walls of the ventricles. So these two are nodes that will emit the electrical activity. The bundle of His and Purkinje fibres are able to transmit and conduct that electrical activity through the myogenic muscle. So the order that this happens then, the SAN releases a wave of depolarization, which means electricity, across the atria. The AVN will then release another wave of depolarization when that first wave of depolarization reaches it. However, there is a layer of non-conductive tissue between the atria and the ventricles, and this prevents that wave of depolarization travelling straight down through the cardiac muscle in the ventricles. That means instead the wave of electricity has to go through the bundle of His and then the Purkinje fibres. As a result, that means the ventricles actually contract at the apex, which is this very tip here, and then the walls of the ventricles contract after a short delay. And the advantage of this is the atria would have fully emptied the blood before the ventricles contract, and also the ventricles contract from the bottom upwards, so you've got a more efficient pumping of that blood out of the heart. Now, the control of that heart rate is where we see the nervous system taking its role. So the medulla in the brain controls the heart rate via the autonomic nervous system. Autonomic meaning it's automatic, there's no conscious thought behind it. So there's two parts. We have a centre link to the sinoatrial node, so the SAN, to increase the heart rate via the sympathetic nervous system. And then there's also another that decreases heart rate via the parasympathetic nervous system. And that's what we can see here. We've got the medulla, and then we've got these two. We've got the parasympathetic nervous system and the sympathetic nervous system. So the heart rate changes in response to pH and blood pressure. And these stimuli are detected by receptors. Chemoreceptors detect changes in pH, and we have pressure receptors, also known as baroreceptors, detect changes in blood pressure. And both of these receptors are found in the aorta and the carotid artery, which is just after the aorta. So the response to pH, the pH of the blood will decrease, meaning more acidic, during times of high respiratory rate or respiration, because if it's aerobic respiration, there'll be lots of carbon dioxide produced, which will dissolve to make carbonic acid, or if it's anaerobic respiration, you're producing lactic acid. Either way, this excess acid needs to be removed from the blood rapidly, because if it's really acidic, it could denature enzymes and proteins as well, such as hemoglobin. So this is achieved by increasing the heart rate so that carbon dioxide will get to the alveoli, diffuse out via gas exchange more rapidly. So that means there needs to be more impulses via the sympathetic nervous system to the SAN to make that natural pacemaker, the SAN, fire more rapidly. The response to pressure is, if the blood pressure is too high, this can cause damage to the walls of the arteries, and it's important that mechanisms are put in place to bring down that blood pressure to prevent that damage. And that results in more impulses via the parasympathetic nervous system to decrease the SAN firing, so the heart rate decreases. Now, if the blood pressure is too low, there may be insufficient supply of oxygenated blood to respiring cells. So in that case, we'd have more impulses via the sympathetic nervous system to increase the heart rate to bring the blood pressure up. So here is the process. The stimulus in this case we've got is an increase in carbon dioxide, meaning lower pH, and an increase in hydrogen ions. Chemoreceptors in the walls of the aorta and the carotid artery will detect that. Then the coordination is more electrical impulses are sent to the medulla, which is the cardiac centre, and that will then result in more impulses sent via the sympathetic nervous system to the SAN to increase the frequency of electrical impulses. The effector is the cardiac muscle, so the SAN tissue, and the response is an increased heart rate because the SAN will be firing more rapidly. Now, for high blood pressure, the stimulus is increased blood pressure. The receptor would be the pressure receptors in the wall of the aorta and the carotid artery, and these stretch when there's high pressure. Coordination would be more electrical impulses sent to the medulla, and then more impulses sent via the parasympathetic nervous system so that the SAM will fire less frequently. The effects is the cardiac muscle, that's in tissues, response, reduced heart rate, and that lowers the blood pressure. Next is 3.6.2, nervous coordination. And here we have a motor neuron, which is actually the only neuron that you need to know the structure of. So we've got the cell body of the neuron, and that contains organelles that you'd find in typical animal cells. So the ribosome, nucleus, mitochondria, for example. And proteins and neurotransmitter chemicals are made there. Then we've got the dendrites, which carry action potentials to surrounding cells. The axon is, we can't see it because it's actually surrounded by the Schwann cell, but it's the long, thin, wire-like structure. And it's a conductive long fibre that carries a nervous impulse along the motor neuron. Schwann cells, so these are these Schwann cells here, and they wrap around the axon to form the myelin sheath, which is a lipid and therefore does not allow charged ions to pass through it. There are gaps in these along the axon, and those gaps are known as the nodes of Ranvier. So we then move on to the concept of an action potential and resting potentials. Resting potentials are when a neuron is not conducting an impulse. So there's not been a stimulus detected. It's just your resting standard state. But even when at rest, there is a difference between the electrical charge inside the neuron, specifically we tend to talk about the axon, and the outside of the axon. And that difference in voltage is our resting potential. So there are more positive ions, so sodium ions and potassium ions, outside compared to inside, and therefore the inside of the neuron is comparatively more negative at minus 70 millivolts, and you do need to know that value. So this is quite a common 2 or 3 mark question to explain how that resting potential is established or maintained. So first of all, there is a sodium potassium ion pump, which involves active transport and therefore ATP. So that's what we can see here in the membrane of the axon. So this co-transporter pump, we have 2 potassium ions being pumped in and 3 sodium ions being pumped out by active transport. That creates an electrochemical gradient. So we end up with more potassium ions inside of the cell and more sodium ions outside of the cell. Because we have this electrochemical gradient, those ions can then move down their concentration gradient. But the membrane is more permeable to potassium ions because there are more of these potassium ion channels, and also there's some that are permanently open, whereas the sodium ion channels, there are fewer of them and some aren't always open. So although you have sodium ions diffusing in, you have far more potassium ions diffusing out, and that's why it's more positive outside than it is inside the axon, and you get this minus 70 millivolts. Just point out that is the minus there. That minus has just gone on to a different row. Now, an action potential is when there is a response to a stimulus, and you only get this response to the stimulus if the stimulus is big enough to increase the neuron's voltage beyond a set point, a threshold. So an increase in voltage or depolarization is due to the neuron membrane becoming more permeable to sodium ions, and therefore it becomes more positive. And once an action potential is generated, it will happen initially at this point, and then it moves along the axon from node to node of Ranvier like a Mexican wave. And we'll come back to that concept once we go through the action potential. So here we can see we've got our resting potential at minus 70 millivolts, but then we have a stimulus. And the stimulus provides energy that can cause the sodium voltage-gated ion channels, so there's voltage-gated sodium ion channels, to open. And if those open, we then start to get sodium ions diffusing into the axon, which increases the positivity inside of the axon. So we can see that it is becoming more positive. When you have the threshold potential reached of minus 55 millivolts, that then causes even more sodium ion channels, but they're voltage gated sodium ion channels, to open. So we get even more depolarization because more sodium ions diffuse in, up to a maximum of positive 40 millivolts. And at that point, when we get to positive 40 millivolts, that causes the voltage gated sodium ion channels to close, and we still have potassium ion channels open. So we've got potassium ion channels permanently open. So you constantly have positive potassium ions moving out. But now at this point, we no longer have positive sodium ions moving in. And that's when repolarization occurs. So it starts to become more negative. And we get even more potassium ion channels opening. So even more positive ions are now moving out and none are moving in. And that's when we get this overshoot, this hyperpolarization going below the resting potential to at least minus 80 millivolts. And this is then when we'd have that resting potential being re-established through active transport with the sodium ion potassium ion co-transporter pump to bring it back to its resting state. So a stimulus has to be large enough to cause the initial voltage gated sodium ion channels to open to reach the threshold. And once that threshold is reached, an action potential will always occur. But if it's a very small stimulus and that isn't enough to open the voltage gated sodium ion channel, then you don't reach the threshold, and it's known as this failed initiation. You don't get an action potential and it goes straight back to resting. Now that is a good thing because we don't want to respond to every single slight change in our environment, so we just overwhelm our senses. We only want to respond to those stimuli that are large enough, that are worthy to respond to, so that it could potentially cause danger and therefore it is worth us responding to it. Now the refractory period is just labelled here, but you actually have the refractory period happening through all of this point when you've reached that action potential, and we'll come back to the refractory period in a minute. But just showing you this concept of the Mexican wave. So that action potential that we just saw here, this curve, will happen at all of these points along the axon at the nodes of Ranvier. So it's like a Mexican wave because we have this increase and that triggers then an increase here, and that triggers it here, here and here. So each time you reach that threshold, that can then trigger an action potential at the next node of Ranvier. So the all or nothing principle, if the depolarization does not exceed the minus 55 millivolt threshold, an action potential does not occur. So we previously talked about that, and that's the nothing part of the principle. Any stimulus that does trigger depolarization to at least that minus 55 millivolt threshold will always trigger an action potential, and it will always peak at the same maximum. Bigger stimuli does not increase the voltage any further. Instead, it increases the frequency at which the action potentials are generated. So that's the all or nothing principle, and it's important because it makes sure that animals only respond to these large enough stimuli rather than responding to every slight change in our environment. The refractory period, so after the action potential has been generated, the membrane enters this refractory period, which is when it cannot be stimulated to go through another action potential. And it's because the sodium ion channels are recovering and can't be opened. So that should say ions there, sodium ion channels. Now this is important because it ensures that discrete or separate impulses are produced. An action potential cannot be generated immediately afterwards, so it has to be separate, and that makes it easier to process the stimulus. It also ensures that the action potential only travels forwards because it can't go backwards because those sodium ion channels are recovering. So it moves forwards, constantly moving its way around the reflex arc or the nervous pathway. And also it limits the number of impulses, and this is important to prevent overreaction to a stimulus. The factors affecting the speed of conductance are myelination and saltatory conduction, axon diameter, and temperature. Myelination and solitary conduction is linked to the importance of these Schwann cells, which create the myelin sheath. So Schwann cells wrap around the axon to form that myelin sheath, which does not allow charged ions or the impulse to pass through it. So that leaves you with gaps where action potentials can be generated, known as the nodes of Ranvier. And as a result, the action potential jumps from node to node, and that is called saltatory conduction. And this is faster because that means you only have to generate action potentials at these nodes of Ranvier rather than every single position along the axon, and that is why it travels faster. A wider diameter will increase the conductance, and that's because it offers less resistance to the flow of ions inside of the axon, so depolarization spreads more quickly. A higher temperature increases the speed of conduction for two reasons. That conductance will be faster because the ions diffuse faster, because they've got more kinetic energy. And also the enzymes involved in respiration will be working faster. So that means there's more ATP for active transport of that sodium ion, potassium ion pump. Synapse is the gaps between the end of the axon of one neuron and the dendrite of another. And that is where the action potential is then transmitted as neurotransmitter that diffuses across the synapse. This here is the whole process of what happens at a synapse. This is worth pausing, screenshot, and you can turn it into a flow diagram, mind map, because this would be a long answer question. So the action potential arrives at the synaptic knob. Depolarization of the synaptic knob causes calcium ion channels to open, and therefore calcium ions diffuse into the synaptic knob. That causes vesicles that contain the neurotransmitter to move towards and fuse with the presynaptic membrane, and that releases the neurotransmitter into the synaptic cleft. That neurotransmitter diffuses down its concentration gradient across the synaptic cleft, and it will bind to receptors on the postsynaptic membrane. When it binds to the receptors on the postsynaptic membrane, that causes sodium ion channels to open. Therefore, sodium ions will diffuse into that postsynaptic membrane of the neuron. And if enough neurotransmitter is bound to cause enough sodium ions to diffuse in to reach that minus 55 millivolt threshold, then it's going to generate that action potential. Lastly, that neurotransmitter needs to be broken down so that it is then released from the receptor to cause the sodium ion channels to close. Because otherwise, even after the stimulus has stopped, that will still be triggering an action potential in the postsynaptic membrane, which we don't want to happen. So that is then going to happen because of enzyme. Enzymes will degrade the neurotransmitter, causing them to be released. Therefore, the sodium ion channels close and it goes back into the resting potential stage. And the neurotransmitter that's been broken down gets recycled and reused. Now, the bits that are in bold and yellow are the parts that I've seen repeatedly in mark schemes that you have to say those terms, and sometimes they're even underlined that you have to use literally that language. So make sure you're really specific with how you word this. A cholinergic synapse is a particular synapse with the neurotransmitter acetylcholine. So it's exactly the process we just went through, except you need to say acetylcholine whenever you would have said neurotransmitter. And you need to know that the enzyme that breaks down the acetylcholine is acetylcholine esterase. And that will break down or hydrolyze the acetylcholine into acetate and choline, which then gets recycled. Summation is what happens at a synapse, and it's the rapid buildup of neurotransmitter in the synapse to help generate an action potential, because we have to have enough neurotransmitter to cause enough sodium ion channels to open for enough sodium ions to diffuse in to trigger and generate that action potential. So the two methods are spatial or temporal summation. Spatial summation is when you have many different presynaptic neurons converging at one postsynaptic neuron. So collectively, these multiple presynaptic neurons release so much neurotransmitter that it should result in the generation of an action potential. Temporal summation is when you just have one presynaptic neuron connecting to one postsynaptic neuron. And in this case, it is the repeated release of neurotransmitter from that one presynaptic neuron over a short period of time that should add up to enough neurotransmitter to cause enough sodium ion channels to open to reach that threshold potential in the postsynaptic neuron. Now, the movement at a synapse is unidirectional. The impulse only travels in one direction. And there are a few reasons why that's the case. Number one is the neurotransmitter is only released by the presynaptic neuron, and therefore you get this concentration gradient. So it can then only diffuse from that location to where there's a lower concentration at the postsynaptic neuron. And secondly, you only have the receptors that the neurotransmitter binds to on the postsynaptic neuron's membrane. You can also get inhibitory synapses, which will prevent an action potential being generated. And these can cause chloride ions to diffuse in instead of or in addition to sodium ion channels. And as a result, because you've got chloride, which is Cl minus, you have negative ions moving in, and that makes that postsynaptic membrane potential even more negative. It could go down to minus 80 millivolts going into hyperpolarization. That makes an action potential highly unlikely because you need even more sodium ions to diffuse in before you'd reach that minus 55 millivolt threshold. A neuromuscular junction is very similar to a synapse, except instead of it being the gap between two synapses, it's actually the end of your nervous pathway where you have a gap between your motor neuron and your effector, which is a muscle. And striated muscle will contract when it receives an impulse from that motor neuron through that neuromuscular junction, and it works in a very similar way to synapses. So first of all, an action potential reaches that presynaptic membrane of the motor neuron, causing calcium ion channels to open, calcium ions diffuse in. That causes the vesicles that contain the neurotransmitter to move towards and fuse with the presynaptic membrane, releasing the neurotransmitter, which is acetylcholine, that diffuses across this gap, and then it will bind to receptors on the postsynaptic membrane. But the postsynaptic membrane here is the sarcolemma, which is a membrane of the muscle fibers. The binding causes sodium ion channels to open in that sarcolemma and sodium ions diffuse in. So it's pretty much identical, except we've got not a postsynaptic neuron, we've got muscle fiber, so that receptors are on the sarcolemma, and it's always going to be acetylcholine as the neurotransmitter. So that depolarizes the membrane of the muscle fiber and creates an action potential in that muscle that moves along these T-tubules towards the center of the muscle fiber. And those action potentials prompt the opening of voltage-gated calcium ion channels in the membranes of the sarcoplasmic reticulum, situated near these T-tubules. And as a result, calcium ions diffuse out of the sarcoplasmic reticulum around the myofibrils. And that is what causes muscle contraction via the sliding filament theory. So here's just a summary table comparing the neuromuscular junction to the cholinergic synapse. So you can screenshot that in case you did get a long answer, compare and contrast question for those. Now this flows into what we were just talking about, muscle contraction. 3.6.3, skeletal muscles and looking at how they can contract. So we talked about the myofibril. Here are the structures that you need to be aware of. Again, this could be a good one to screenshot, turn into flashcards for each of these structures. The myofibril, these are long cylindrical structures found inside muscle fibers made up of repeating sarcomere units, which we're going to have a look at. Because the sarcomere is the functional unit. It's defined as a region between two Z lines, and you have actin and myosin where you can have your sliding filament theory occurring. The sarcolemma, that's the plasma membrane of a muscle fibre. It conducts electrical impulses, the action potentials, and it triggers the contraction to help maintain ion balance within the cell as well. Sarcoplasmic reticulum, that's a specialised form of smooth endoplasmic reticulum that surrounds myofibrils, and it stores and releases calcium ions, which are essential for triggering muscle contraction. The sarcoplasm, that is the cytoplasm of a muscle fibre. So it contains the organelles, enzymes, myoglobin needed for energy release and muscle function, and it also has glycogen stores for ATP synthesis. So looking at the sarcomere in a bit more detail, this is what we said on the previous slide. So this is one long cylinder, which is the myofibril, made up of these repeating units called sarcomeres, which are identified by two Z lines. There are other structures within the sarcomere that you need to be able to name. So here are the Z lines, which indicate the start and end of one sarcomere. The A band is the myosin. So that is the distance of myosin, which is shown in blue here. An I band is the distance where you have actin not overlapped at any point with the myosin. So just red lines here, which are our actin protein. We have an M line, which is the center of where the myosin is attached. We have an H zone, which is where you just have myosin without any actin overlapping it. So those are your key structures, all to do with basically whether you have actin by itself, myosin by itself, and where it's overlapping. And the kinds of questions you might get is, what happens to the size of those sections when a muscle contracts? And when a muscle contracts, the actin slides inwards over the myosin, which we can see here. So as a result, the H zone decreases in length because we now have less myosin by itself. The I band will also decrease in length because we have less actin by itself because it's slid over. The A band never changes because that is the length of the myosin. Myosin doesn't change in length. The M line will always stay in the same position as well, but the Z lines move closer together. So that's something you need to know as well, those different bands and what happens to them when a muscle contracts. The sliding filament theory explains why you have the actin sliding over the myosin. So when that action potential reaches a muscle fibre, like we saw in the neuromuscular junction, it triggers the release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm. And those calcium ions cause tropomyosin, which is a protein that is normally blocking the binding sites on actin, to move, and it exposes the binding sites for myosin on actin. Now, because of that, we get this cross bridge formation. So with ADP and PI still attached to our myosin head, which is a structure branching off myosin, that can now bind to the myosin binding sites on actin. And when that happens, it creates what's known as an actino-myosin cross bridge. The power stroke, which is when the actin filament is moved, once that cross bridge is formed, the myosin head pivots, so it bends here, and that pulls the actin filament inwards, so it slides it in. And that movement is known as the power stroke, and when that happens, it causes the release of ADP and PI. It also generates tension, and that is causing the actin filament to slide. So we then get this ATP molecule binding, and that new ATP molecule, when it binds to the myosin head, it causes it to detach from the actin filament. And this ensures that the myosin head doesn't remain permanently attached to the actin. That ATP then gets hydrolyzed, and it resets the myosin head. So within the sarcoplasm, there is the enzyme ATPase, and that is activated by calcium ions. That will hydrolyze the ATP back into ADP and PI, And the energy released from that ATP hydrolysis repositions the myosin head back to its original state, ready to form a new cross bridge and therefore go through that process again to bring the actin even closer for a full muscle contraction. So as long as calcium ion levels remain high, the cycle repeats, continuously pulling the actin filaments towards the center of the sarcomere, shortening the muscle fiber and causing contraction. When the nervous system stimulation stops, calcium ions are then actively transported back into the sarcoplasmic reticulum, and that allows the tropomyosin to move back to block the myosin binding sites on actin to prevent any further contraction, and the muscle will relax. So some common questions. What's the role of ATP and phosphocreatine? Here are your standard answers if this came up. So the ATP is hydrolyzed and the energy will then break the actinomyosin bridges, or it's to move or bend the myosin head back to its original state, and for the active transport of those calcium ions. So if ATP levels are low, muscle contraction can't happen effectively. And that is where phosphocreatine plays a role, because this acts as a rapid source of ATP in muscle cells by donating a phosphate group to ADP to be able to make ATP, so that you do have ATP during short bursts of intense activity. You could also be asked what's the role of glycogen granules in skeletal muscles. So it's a store of glucose that can be hydrolyzed into glucose, and that glucose can be used as a respiratory substrate to provide the ATP needed. The role of calcium ions then, so when an action potential reaches the muscle fibre, calcium ions are released from the sarcoplasmic reticulum and diffuse into the myofibrils. Those calcium ions cause tropomyosin to move and expose the binding sites on the actin filament. And with these sites now accessible, the myosin heads attach to actin, forming the actinomyosin cross bridge. The last bit of this topic is knowing the difference between slow twitch muscle fibres and fast twitch muscle fibres. So knowing the difference between the structure, location, general properties, the type of respiration that happens, ATP production and energy storage. So I recommend screenshot this page, or if you've downloaded my free booklet, fill it in so you've got this for your revision. So in terms of structures, slow twitch, large store of myoglobin, rich blood supply with many mitochondria, compared to fast twitch, thicker with more myosin filaments, large store of glycogen and a store of phosphocreatine. Location, you get a lot of slow twitch in calf muscles, whereas fast twitch might be places like biceps. General properties, slow twitch contract slower, and they can sustain aerobic respiration for longer periods. So these muscles are used in endurance activities like marathons. Fast twitch contract faster and provide short bursts of powerful contraction for things like sprinting or weightlifting. Slow twitch will mainly be aerobic, fast twitch is anaerobic, and in terms of ATP production, the slow twitch produce ATP slowly, but they'll generate more, and that's because it's aerobic. And because fast twitch are anaerobic, they produce ATP quickly, but less. Lastly, in terms of the glycogen store, they both have stores of glycogen, but slow twitch will have a lower store of glycogen, and you'll have a large store in the fast twitch. Next then is 3.6.4, homeostasis. Homeostasis involves physiological control systems that maintain the internal environment within restricted limits. And it's important in maintaining things like a stable body temperature, stable blood pH for enzyme activity. Negative feedback is involved, and this restores systems to their original level. And the possession of having separate mechanisms, so one that can bring levels back up, one that can bring it back down, give better control to get it back to the original state that it should be at. So for body temperature, if it's too low, there'd be insufficient kinetic energy for enzyme controlled reactions. If it was too high, enzymes would denature. Either way, metabolic reactions would stop or slow and cells would die. If you have too high or too low a blood pH, it will result in enzymes denaturing, fewer enzyme substrate complexes, and again, that are going to affect metabolism. Blood glucose control. So glucose is needed for respiration. So a lack of glucose in the blood could result in cell death. And if blood glucose levels are too high, then this lowers the water potential of the blood, and water then leaves the surrounding cells by osmosis, and that prevents the normal function of those cells because now they're low on water. And also it increases the blood pressure because there's more liquid in the blood. The water potential of the blood then, if it's too high, water moves into cells by osmosis, and that could cause them to burst. And if the water potential is too negative, the water leaves the surrounding cells into the blood, and that can cause the cells to crenate. So you need to know in detail blood glucose concentration. And that is going to increase after you've eaten or drunk something containing carbohydrates, or it will decrease if you haven't eaten something containing carbohydrates or following exercise, because respiration rates would be higher using up your carbohydrates. Here are the different things involved. So the pancreas, that is the organ that detects changes in blood glucose levels. And specifically, you have the islets of Langerhans cells that are going to detect and release insulin and glucagon to bring blood glucose levels back to normal. Insulin is a protein hormone released when blood glucose levels are too high, so it causes a decrease in blood glucose levels. Glucagon is also a protein hormone, and it's released when blood glucose levels are too low, and that will cause an increase in blood glucose levels. Now, adrenaline, this is released by the adrenal glands when your body anticipates danger. So it's not to do with homeostasis, but it has a very similar effect to glucagon because it causes glycogen to be hydrolyzed, so you increase the blood glucose levels. So some key terms that you need to know for this topic are glycogenesis, glycogenolysis, gluconeogenesis. So learn those terms, get them on a flashcard. Wherever you see glyco, that means it's about glycogen. So these top two are about glycogen. Where you see gluco, that means it's about glucose. Genesis means to make, lysis means to break. So glycogenesis means making glycogen. So it's the process when excess glucose is converted into glycogen. And that happens in the liver quite a lot and muscle cells. Glycogenolysis, so glycogen lysis is the hydrolysis of glycogen into glucose. Gluconeogenesis. Gluco means glucose, genesis means to make, neo means new. So it's making glucose from something new. And by that we mean a non-carbohydrate source. So for example, from amino acids, or it could be from glycerol from lipids. So your normal blood glucose levels might increase if you've just eaten a carbohydrate-rich meal. That is detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will then release insulin. That causes liver cells to become more permeable to glucose, so more glucose is taken from the blood into the liver cells, and then that glucose is converted into insoluble stores of glycogen. That means that we've removed glucose from the blood and brought our blood glucose levels back to normal. Now, maybe you haven't eaten anything containing carbohydrates for a while or you've done high levels of exercise, so lots of respiration using up the blood glucose levels, so they decrease. A decrease is detected by the alpha cells in the islets of Langerhans of the pancreas. In response, the alpha cells release glucagon. That triggers the second messenger model, which activates enzymes to hydrolyze glycogen in the liver back into glucose, which is then released into the blood to bring it back to normal. But we're going to go through that in a bit more detail. So here's our action of insulin. We said that's the hormone released by the beta cells in the islets of Langerhans when blood glucose levels are too high. And insulin will then be transported in the blood. When it gets to the liver cells, it will bind to complementary shaped receptors on the cell surface membrane of those liver cells, also muscle cells. When it binds, it increases the permeability of that membrane to glucose by stimulating the insertion of more glucose channel proteins, GLUT4, into the membrane. And that allows more glucose to move from the blood into the liver cells by facilitated diffusion. Now, once that happens, the glucose has to be converted to glycogen because that is insoluble and therefore it's not going to lower the water potential of the cell like glucose would, and therefore it won't affect osmosis. So these actions help to reduce the blood glucose levels back to normal via negative feedback. And this is just showing us here when the insulin binds to complementary shaped insulin receptors embedded within the membrane of the target cell, the liver cell. That causes a cascade of chemical reactions, which then causes vesicles, which contain the glucose channel protein, to move up and fuse with the cell surface membrane, and it embeds more of those channel proteins. So more glucose can then move from the blood into the cell. The action of glucagon then, the alpha cells in the islets of Langerhans detect the blood glucose level is too low and secrete glucagon, which will travel around in the blood, and when it gets to the complementary shaped receptors on the cell surface membrane of liver cells, it activates a second messenger model, which is described here. So it causes a protein to be activated into adenylate cyclase. That is converting ATP into cyclic or cyclic AMP. Cyclic AMP activates another enzyme, protein kinase, and that's the one that hydrolyzes glycogen into glucose to release it back into the blood. Now, if your glycogen stores are used up, then you can also have the activation of enzymes that can convert glycerol and amino acids into glucose to be used as a respiratory substrate instead. So here's our second messenger model. The first messenger is glucagon. That hormone binds and it activates enzyme number 1, which is adenylate cyclase. Now, adenylate cyclase is going to convert ATP into cyclic AMP, which is our second messenger. And that will then activate the protein kinase, which can hydrolyze glycogen into glucose. Now, adrenaline also goes through that same second messenger model, so you get that same process happening. Diabetes, you have type 1 and type 2 diabetes. Type 1 is when you're unable to produce insulin. Usually starts in childhood, and it could be a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections or some way of getting insulin into the blood. Type 2 diabetes is when the receptors on the target cells lose their responsiveness to insulin. So insulin is still made, it's just the receptors don't respond as they should. And this usually develops in adults because of obesity and poor diet. It can be controlled by regulating the intake of carbohydrates, increasing exercise to use up that glucose for respiration, and sometimes insulin injections are still needed as well. Osmoregulation. So this is now looking at the regulation of the water potential of the blood. And blood with too low a water potential is described as hypertonic. And that might be if you've been sweating lots, not drunk enough, or there's lots of ions in your diet. Hypotonic is if the blood has too high a water potential, and maybe you've drunk too much water or you don't have enough salt in your diet. The corrective mechanisms would be, if you've got too low a water potential, then more water is reabsorbed by osmosis into the blood from the tubules of the nephrons in the kidneys. So that would mean you make urine, which is more concentrated and lower volumes. If you've got too much water in your blood, so too high a water potential, then less water is going to be reabsorbed by osmosis back into the blood from the nephrons. So you end up with larger volumes of dilute urine. So let's have a look at this nephron then. Osmoregulation occurs within the nephrons in the kidneys, and nephrons are these long tubules surrounded by capillaries, and there are around 1 million per kidney. The blood gets filtered here to remove waste and selectively reabsorb useful substances back into the blood. So the key stages, which we will go through each in more detail, we have ultrafiltration first, and that occurs due to high hydrostatic pressure. That will force out from the blood water and small molecules like glucose. And that gets forced from the glomerulus capillaries into this renal capsule. Then in the proximal convoluted tubule, we have selective reabsorption occurring. And that is where we're going to have all the glucose and some water being reabsorbed. Then we've got the loop of Henle, and this maintains a sodium ion gradient, so water can be reabsorbed into the blood. And water then will continue to move out of the distal convoluted tubule and collecting duct by osmosis, and the collecting duct carries the remaining liquid, which is the urine, to the ureter. So the structure of the nephron then, the formation of the glomerular ultrafiltration, that occurs here in the renal or Bowman's capsule. And there is a high hydrostatic pressure in these capillaries, the glomerulus. And as we said, that forces out water, glucose, ions and urea from the blood into that capsule. Large molecules like proteins and red blood cells remain in the blood. So here we have what causes that high hydrostatic pressure first of all. As the blood enters the glomerulus via the afferent arteriole, which is a wider blood vessel, the difference in that size creates a high pressure inside the glomerular capillaries, and it's the high hydrostatic pressure that forces out all of those small solutes and the water. Now you do have 3 filtration barriers. We first of all have the capillary endothelium. So the glomerular capillaries have fenestrations, which are these tiny gaps between the endothelial cells. And that will allow things like plasma and dissolved substances to pass through, but it blocks the red blood cells. So by plasma, I mean it'd be like the water, urea, and glucose amino acids, but blood cells can't fit through there. The next layer is the basement membrane. And this is a gel-like protein-rich layer between the capillary and the Bowman's capsule. It acts as the main filtration barrier and it prevents the passage of the large proteins. So small molecules can pass, but large proteins cannot. And then finally, we have podocytes. And podocytes have foot-like extensions that wrap around the capillaries that create these gaps, these filtration slits, to further ensure only small molecules can enter. So that's how we have the filtration. Then we have the selective reabsorption. So all glucose is reabsorbed by active and co-transport, and most water is reabsorbed by osmosis at this stage. The proximal convoluted tubule, which it says PCT here, do not abbreviate that in the exam. It is not an accepted abbreviation. The proximal convoluted tubule has cells have many microvilli and mitochondria to maximize this active transport and co-transport. So those cells lining the proximal convoluted tubule actively transport sodium ions out of the epithelial cells into the blood via sodium-potassium pump. That creates a lower concentration of sodium ions in the proximal convoluted tubule compared to the lumen. And therefore sodium ions diffuse into the proximal convoluted tubule down their concentration gradient, but that has to be via a co-transporter protein which carries glucose with it as well. So that's how we get the glucose inside of the proximal convoluted tubule, epithelial cells. And then once inside there, the glucose moves by facilitated diffusion into the blood, and that is how all of the glucose re-enters the bloodstream. So adaptations include these cells have microvilli, so these highly folded cell surface membranes to increase the surface area for that transport. They also contain many mitochondria so that lots of ATP can be produced for this active transport. And then there'll also be lots of carrier proteins and co-transporter proteins embedded in the cell surface membranes for all of that transport. And then water is reabsorbed by osmosis because following the absorption of the solutes, that creates a water potential gradient, then water can move in by osmosis as well. Then we get to the loop of Henle where we have the maintaining the sodium ion gradient. And in the descending limb, water leaves by osmosis, but sodium ions cannot. So the descending one is the one going down. The ascending limb is the one going up. Sodium ions are actively transported out, but water cannot leave. And this creates a water potential gradient in the medulla, so this surrounding area outside of the nephron, which allows even more water to be reabsorbed by osmosis at the distal convoluted tubule and the collecting duct. And water reabsorption is regulated by the hormone ADH, which we're going to come on to shortly. So here's a bit about the loop of Henle in a bit more detail, because we said that descending limb, the walls are thin and permeable to water, but impermeable to ions. So as the filtrate moves down the descending limb, water will move out by osmosis into the surrounding medulla due to that lower water potential. and the concentration of the filtrate will increase. As it moves up, the filtrate moves up the ascending limb. The walls are thicker and they're also impermeable because they don't have aquaporins within them, so water cannot move out by osmosis. But sodium ions can be actively transported out, and that then means we get a really negative water potential gradient. Now at the base of the ascending limb, where the filtrate is very concentrated, some sodium ions will also move out by facilitated diffusion. But this whole process creates a high concentration of sodium ions in the medulla, lowering the water potential, which is essential for water reabsorption in the distal convoluted tubule and collecting duct. So how the hypothalamus and the posterior pituitary gland are involved. The hypothalamus is going to detect changes in the water potential. Specifically, we have osmoreceptor cells, which are the receptors that detect the changes in water potential. So if the water potential of the blood is too low, water is going to leave the osmoreceptors that are in the hypothalamus, causing those cells to shrivel, and that stimulates the hypothalamus to produce more of the hormone ADH, antidiuretic hormone. And if the water potential of the blood is too high, water enters the osmoreceptor cells by osmosis, and that stimulates the hypothalamus to produce less ADH. Now, the hypothalamus is where ADH is produced, but ADH then moves to the posterior pituitary, and that is where it is released from into the blood. You do need to know that distinguishment. It's come up as an exam question before. And once that ADH is released in the blood, it then travels in the blood to its target organ, which is the kidney. So ADH, antidiuretic hormone, this is going to cause an increase in the permeability of the walls of the collecting duct and the convoluted tubule. And as a result, more water is going to move from the nephron by osmosis back into the blood. And that results in urine that is more concentrated and in smaller volumes. So there are receptors on the cell membranes of the distal convoluted tubule and the collecting duct to which ADH bind. When ADH reaches the kidney, it binds to those receptors. And when bound to the receptors, it activates a phosphorylase enzyme in the cells. The phosphorylase causes the vesicles containing aquaporins to move and fuse with the cell membrane, so you get more of these aquaporins, which are protein channels that water can move through. Now, this is a little bit extra information. You could include it in an essay. You could still get marks sometimes in the mark scheme for it, but it's not essential that you need to know that process. You do need to know that ADH binds to the receptors, increases the permeability of the cell membrane to water. Aquaporins is often a mark as well. So therefore, ADH causes an increase in the permeability of the walls of the collecting ducts and the convoluted tubule, which means more water will move into those cells and then into the blood by osmosis. And therefore, urine is more concentrated and you return more water to the blood. So in summary then, if the water potential of the blood increases, meaning too much water, so you might have drunk a lot of water, that's detected by the osmoreceptors in the hypothalamus. Hypothalamus produces less ADH. The distal convoluted tubule, which again, that is not an accepted an abbreviation. Do not abbreviate it like that in the exam. You have to write it out in full. ADH you can abbreviate though. So the distal convoluted tubule and the collecting duct walls become less permeable to water. So that means less water is reabsorbed in the blood and more remains in the filtrate in that collecting duct and goes on to create larger volumes of more dilute urine, bringing your water potential of the blood back to normal. Now, maybe you've sweat a lot and you've lost a lot of water and your water potential of the blood decreases. That is going to be detected by the osmoreceptors in the hypothalamus. The hypothalamus will produce more ADH, which is released into the blood by the posterior pituitary. The distal convoluted tubule and the collecting duct walls become more permeable to water, so more water is reabsorbed into the blood by osmosis and less is lost in the urine. So you get more concentrated urine, and that means you get your normal water potential of the blood. AQA topic 7. And it starts with 3.71, inheritance for A-level only. There are lots of key terms in this topic that you could be asked for a 1 or 2 mark question. So I recommend that you screenshot this or when the answers are there, or turn this into flashcards because these are essential to learn. Genotype, that's the genetic constitution of an organism, which basically means the alleles it has for a particular gene. Phenotype is often 2 marks because it's the expression of the genetic constitution for 1 mark. Second mark is interaction with the environment. Homozygous and heterozygous I haven't seen come up as definition questions, but it's important that you know what those mean. Homozygous meaning you've got 2 alleles that are the same for a particular gene. Heterozygous is 2 different alleles for the same gene. Recessive allele and dominant allele have both been assessed before. Recessive is an allele that's only expressed if there is not a dominant allele present, whereas a dominant allele will always be expressed in the phenotype. Codominant is when both alleles for a gene are equally dominant and are both expressed in the phenotype. Multiple alleles is if there are more than 2 alleles for a single gene. Sex linkage is when the gene of interest is located on the X chromosome. So locus means the location of the gene on a chromosome. Autosomal linkage is when genes are located on the same chromosome. So it's going to be 2 different genes are found on the same chromosome, but it's not the sex chromosomes. Epistasis is when one gene modifies or masks the expression of a different gene at a different locus. Monohybrid and dihybrid don't come up as definition questions, or at least they haven't, but it's just knowing what they mean. So monohybrid, mono meaning 1, it's looking at the inheritance of just 1 gene. A dihybrid cross would be looking at the inheritance of 2 genes. So inheritance then, we're going to go through all of the types that you need to know for AQA, but there's different ways to present what allele you have for a particular gene. And that's what I mean here by this coding. This is what we'll use when we do Punnett squares or flow diagrams to work out predictions of what you might see in offspring. So for a simple monohybrid cross, that's what you would have done at GCSE. There's just 1 gene with 2 alleles, the dominant or recessive. So you pick 1 letter to represent the gene. Capital is dominant, lowercase is recessive. But then when we get to codominance and multiple alleles, we need to do the coding differently. Because for codominance, it means both of the alleles are dominant. So we can't pick 1 letter and have it capital for dominant because they're both dominant. So instead, you pick a base letter to represent the gene. And then superscript, you have the 2 alleles. So a dominant B, dominant A. And it's not going to be the same letter because they have to be capitals to show their dominance. Multiple alleles, because it's more than 2 alleles for a gene, again, you can't pick 1 letter because that only gives you 2 versions. So we use the base letter to represent the gene. Superscript, you pick the allele letter. And for all of these, if they haven't told you what letters to use, you can pick any letters that you want. But I recommend that you pick letters where the lowercase and capital letter are completely different shapes, like all the examples I've done here, so that it couldn't be confused by an examiner whether you did put the capital or lowercase. Sex linkage, you have the base letter as the chromosome, so whether it's the X or the Y chromosome, and the allele is superscript next to that. There won't ever be anything written next to the Y chromosome because you only get the gene on the X chromosome. Autosomal linkage, this is where we said you have 2 different genes on the same chromosome. And usually this is just done with simple monohybrid inheritance. So 1 letter to represent the gene and then capital and lowercase for dominant and recessive, and that's for both genes. Same with epistasis. This is when 1 gene masks or modifies the expression of the second gene. So we've got 2 different letters representing those 2 different genes, and then dominant and recessive is shown by capital or non-capital letter. So let's have a look at some examples, starting with monohybrid inheritance. Cystic fibrosis is caused by a recessive allele. If 2 carriers reproduce, what's the probability they'll have a child with cystic fibrosis? And on top of that, what's the probability they will have a girl with cystic fibrosis? Now you can pause, have a go. I'm going to go straight through. One thing to be really aware on these questions is they're doing this more often now, where they specify the sex of the child as well, when it isn't a sex-linked inheritance question. So what that means is we need to do our Punnett square to work out the probability of having a child with cystic fibrosis. And it's always 50% probability of having a girl, 50% probability of having a boy. So whatever your answer is for the cystic fibrosis, you then half that because that's 50%. So here are our two carriers. I've picked the letter F here because the capital and lowercase are different shapes. We could pick A. That's even more obvious if you had a capital A, lowercase a. So we can see here that on our Punnett square, if those are our two parents' genotypes, I've then put in the possible gametes that each parent could produce. I've then done our cross to show F with this F, the two capitals, lowercase and capital, lowercase and lowercase, and capital and lowercase. And because cystic fibrosis is caused by a recessive allele, they will only have cystic fibrosis if they are homozygous recessive, because there's no dominant alleles there in their genotype. So we have 25% probability that these two parents will have a child with cystic fibrosis. Then we need to also account for a girl with cystic fibrosis. We need to half that. So times that by 50% or half it is 12.5%. Next, we have a codominant example. Cows can be red, white or roan in colour. Red and white are both dominant. If two roan cows reproduce, what's the probability they will have offspring that are red? Again, pause now and you can have a go at this Punnett square. I'm going to go straight through it. So for this one, parental genotypes, red and white or roan. Red and white are both dominant. So that means those are our two dominant alleles. Now, they'll probably give you more information than that in a question to make it explicitly clear that roan isn't a third allele. They'd need to either tell you it was codominant or give you a hint towards that a bit more. So here, red and white are both dominant. And when red and white are the codominant genotype, you get roan. This is roan here, is when you've got both the red and the white being expressed in the phenotype. And yes, I would personally say that's more brown than red, but it's called red for some reason. So there's our two parents who are both heterozygous. They have that codominant genotype. So if we do our Punnett square, both parents would have one of the white alleles and one of the red alleles. We then combine those in our Punnett square, and we can see we'd have roan, white, roan, and this would be our only one that would be red. So 25% probability of having a red cow. Now, when you do these Punnett squares or flow diagram, it's really important that you are showing the parental genotype if that's not already been given. Then the gametes, and the difference between the two is the gametes are separating those two alleles, whereas the genotype, they're both written next to each other. You then would have to show the offspring genotype and the matching or corresponding phenotype for that genotype. That's why I always leave a space and write the phenotype in the box so it's matched, and then we've got our probability. So then we've got a blood group example, and this is not on the spec. It's just a common example because it models codominance and multiple alleles. So blood groups, you could be blood group A, B, AB or O, and this is referring to the type of antigen you have on the outside of your red blood cells. So some people have antigen A, some have antigen B, some have both antigen A and B, and some have no antigens at all. So that's what the blood groups are corresponding to. And the genotypes for this are, to be blood group A, you could either have two copies of the dominant A allele, or one copy of the dominant A and one copy of O, because O, blood group O, that recessive allele, well, it is a recessive allele. Blood group B, so you could have two copies of dominant B or B and the recessive O. Because O is recessive, you are only going to be blood group O if you have two recessive O alleles. And then blood group AB is the codominant genotype, because if you have A and B, they're both dominant, they are both expressed in the phenotype. So we've got codominance and multiple alleles because we have three alleles. So if we have a look at a genetic cross example, as I said, this is not on the spec that you need to know blood groups. So they would have to give you information about it to be able to work it out. But I went through it because it's a relatively common example, or at least it used to be. So we're told we've got parents with blood group AB and blood group O reproducing together. In the question, we're then told IA and IB are codominant and IO is recessive. What is the probability that they will have an offspring with blood group A? So the first step is working out the parents' genotypes. And if they are AB, they must be A and B, that codominant. And if it's O, that is recessive, so homozygous recessive for O. Then we can put in the parents' gametes. Then we can do the cross, and then we can add in the blood group, so the phenotype corresponding to those genotypes of the offspring. And we were asked the probability of having an offspring with blood group A, and that is 50%. Then we've got a sex linkage example. So colour blindness, again, you don't need to know this as an example. In fact, all of the examples I've done are application. There is no genetic condition or disease that you need to know for AQA. You'd always be given the information and then you do the genetic cross. So colour blindness is caused by a recessive allele, or at least one type of colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be colourblind? So step one is always working out the genotypes of the parents. And when it's sex-linked, you get marks for including the sex chromosomes in the genotype as well. So you do have to include that. For the male, it's XY. For the female, it's XX. And remember, you only find the allele on the X chromosome. So the female here is a carrier, and we're told it's recessive. So there'd be X. I've picked the letter R, so it's really clear, the dominant versus recessive. So X, capital R, lowercase r. And that's a times, that's not part of the genotype. That is crossed with the male, which is X, capital R, Y. Because they are not colourblind, they only have the dominant allele. So then if we have a look at our cross, you might have noticed it's actually slightly changed. So I just went and edited it to really emphasise a key point, which I'll get to. So we have the male gametes here, X, capital R, and then the other gamete would just be the Y. Then we've got the X, lowercase r, and X, capital R for the female, the mother. Now, I've actually done that the other way round, how I've written it here, because there it was the capital first, but it doesn't matter. Your answer ends up the same. So we then fuse together the gametes to show all of the possible genotypes in the offspring. And then I've added the corresponding phenotypes for those genotypes. And you have to state the sex as well as the characteristic linked to the gene of interest. So here it's female, normal vision. They may actually call that colour vision. In the older exam questions, they used to say normal, but they've started to not use that now because it implies if you don't have colour vision, then it's not normal. So they would say colour vision or normal vision. But it's female and they have colour vision. Female colour vision, male colour vision, male colour blind. So you have to state the sex and also the characteristic in the phenotype. And for this one, that's 25% probability of having a colour blind child. Epistasis then, that was when one gene influences the expression of another. And this is often seen in colours. So it could be coat colour in mice. It could be coat colour in Labradors. It could be fruit colour of vegetables. So these are the common examples that you see. And we're going to go through an example and then do the cross. So let's have a look at coat colour in Labradors. The first gene controls whether pigment is going to be produced. And if you have the dominant version of that gene, so dominant allele, then that codes for pigment will be made. The recessive version codes for no pigment. So if a dog is homozygous recessive for this gene, they will not produce any pigment, and that's how you get the yellow dog colour. So that is no pigment has been produced in the fur. Gene 2 controls which colour pigment is expressed if they are able to make pigment. Capital B is black fur, lowercase b is brown fur. So that's how we get these three different colour furs, or hairs if it's dogs, from 2 genes. So let's have a look at the cross here. We are told straight away the parents' genotypes. And you can pause now and have a go at first we're working out what would the phenotype be of these 2 dogs. So we've got capital E, lowercase e. This was the one that controls where the pigment is made. This is the gene that controls what colour that pigment is. And for this one, they'd both be black because they've got the capital E, which means pigment can be made. Capital B means the pigment would be black. So if we then did a genetic cross, first of all, we have to work out all of the possible gametes both dogs could make. And because this is now a dihybrid cross, meaning the inheritance of 2 genes simultaneously we're looking at, you have to show all the possible combinations that you could get. So you need to show just 1 copy of each gene. So 1 letter E, 1 letter B, but all of the possible combinations. So we could have the capital E with the capital B, the capital E with the lowercase b, the lowercase e with the capital B, the lowercase e with the lowercase b. So we get 4 possible gametes for both of those parents. So that means our Punnett square is going to be 4 by 4. It's much bigger. And we now need to show all of the possible genotypes. And I've then added in all of the possible phenotypes as well. So here we go. We can see all of those genotypes and the corresponding phenotype. And we have 9 black, 4 yellow, 3 brown. So we've got a 9:4:3 ratio for this. So that actually goes into dihybrid crosses. A dihybrid cross is the inheritance of 2 genes at the same time. And we've just done one example, but here is a more straightforward example where we don't have epistasis involved. So we've got a round and yellow pea, a wrinkled and green pea. So 1 gene is coding for shape, 1 gene is coding for colour. And with all genetic crosses, we need to have the parent's phenotype, the parent's genotype, the possible gametes the parents could make, the offspring genotype, the offspring phenotype, and then the proportion of each phenotype. Sometimes 1 and 2 might be given to you, sometimes they aren't. So for this one, the genotypes, you would need to have information to know this, but we've got round and yellow, and in this instance, both parents are homozygous for both genes. They would have needed to tell you that for this parent, you wouldn't know otherwise, because it's a dominant characteristic, round and yellow. So they could have either been homozygous dominant or heterozygous for both genes. So round and yellow, wrinkled and green are the 2 recessive characteristics. They had to be homozygous recessive for both genes. Possible gametes then, the round and yellow can only give dominant alleles. So 1 dominant R, 1 dominant Y in every gamete. And this one here can only give recessive alleles. So all of the gametes would have 1 recessive R, 1 recessive Y. So that means we could do a Punnett square, but actually every offspring will get this gamete and this one. So combining them together, that gives us offspring that are heterozygous for both genes. And that would mean they'd be round and yellow. So 100% are round and yellow in our first generation in this dihybrid cross with these parents. Now you could be asked to then cross F1, which means first generation. And that would now mean we're crossing 2 peas that are heterozygous for both genes. And this is now when we'd get 4 possible gametes for both of those genes. So capital R with the capital Y, capital R with the lowercase y, lowercase r with the capital Y, lowercase r with the lowercase y. So looking at all the possible combinations of getting just 1 of the R's and 1 of the Y's in every gamete. And this is then when you get that big Punnett square again, the 4 by 4 Punnett square. And if we combine all of those together and then add in all of the phenotypes to the corresponding genotype, we can see we get a 9:3 to 3:1 ratio. And in a dihybrid cross, if you have 2 parents that are both heterozygous for both genes, like we saw here, then you will always get 9:3:3:1 because you'll always have this pattern of gametes. Therefore, crossing it together, it doesn't matter what the genes are, you'll still get this outcome. So sometimes for 1 mark, you don't have to do the Punnett square and they just ask you and they tell you, you've got a dihybrid cross. both parents are heterozygous for both genes, what will the ratio of the phenotypes be? And it's just 9 to 3 to 3 to 1 because it's learning that fact. However, there are exceptions. So if there is autosomal linkage, you won't get that ratio. If there's crossing over in meiosis, you won't get that ratio. And if there's epistasis, you don't get that ratio. So sometimes that is also asked as just a 2-mark question. Maybe it's to give 2 reasons why you might not get that, or suggest 2 reasons why you won't get that ratio. And it's those 3 reasons. And that leads us into what is autosomal linkage and how does it mean, or how does it result in you getting a different ratio? Now, I'm actually just going to quickly go over crossing over in meiosis very briefly, because this actually is from topic 4, but you need to have an awareness of it for this. So crossing over of homologous chromosomes happens in meiosis 1, the first round of division in meiosis. It's when those homologous pairs, so the chromosomes, pair up at the equator to form a bivalent, meaning 2 chromosomes next to each other. The non-sister chromatids can cross over, get tangled, break and swap that section of the chromatid. And as a result, when you come to work out what gametes a parent might be able to produce, you get new combinations of alleles in the gametes. So that's the relevance here, because you might have thought, well, these parents could make these 2 alleles, but if crossing over happens, maybe they can actually make 4 different gametes. So autosomal linkage, this is when we have 2 different genes located on the same chromosome. And as a result, they have to be inherited together. So it's going to impact the gamete predictions, because I'm using the same example here. The 2 Ps, heterozygous for both genes, the capital R, lowercase r, capital Y, lowercase y. But now we're imagining that there's autosomal linkage. So we've got gene 2, which was for shape, gene 1 is for colour. They're still heterozygous for both genes, but we can see this chromosome has the dominant R and the dominant Y. This chromosome of the pair has the recessive R and the recessive Y. And that means after meiosis, you will have to have gametes that are capital R, capital Y, because this chromosome will be going into one gamete, this one will be going into the other. So we previously said when there was no autosomal linkage, there were 4 possible gametes that parent could make, but with autosomal linkage, there's actually only 2 possible gametes that these parents could make. So here was our original when there was no autosomal linkage, heterozygous for both genes, and here are all the possible gametes. So these 4 and these 4 for that parent. But now there is autosomal linkage, we can actually only create these 2 gametes. And that's the case for both parents because they both have this autosomal linkage. So that now means that this is the possible genotype and phenotype combinations we could get. So we have 3 yellow and round and 1 green and wrinkled, which is not our 9 to 3 to 3 to 1 ratio. So if there is autosomal linkage, that is how it's going to affect the ratio. But now let's add in though, crossing over as well. So here's what we've said so far. With these 2 parents, we would expect 9 to 3 to 3 to 1 ratio. But if there's autosomal linkage, we actually had a 3 to 1 ratio. However, sometimes you're told that information and then you say, scientists actually observed the following results. And we can see we have 4 phenotypes, and here are the numbers they observed. They've got mainly yellow and round, then green and wrinkled, and then you don't have very many yellow, wrinkled or green round. And they would ask you, how is it possible they got these results? And the only way this is possible is if there is autosomal linkage and crossing over in meiosis, because that crossing over in meiosis created new combinations of alleles in the gametes, and that's why it was possible to get these 2 extra phenotypes. So let me show you a common answer for that. This is actually just modelling what we mean here. So here we had our autosomal linkage. We've got the recessive alleles for those 2 genes on this chromosome, the dominant on this chromosome. However, there was crossing over in meiosis 1. Those chromatids break off and swap. So as a result, we now have new combinations of alleles in the gametes. This was the original, so the non-recombinant, the 2 recessive alleles. This one at the end is also non-recombinant, meaning it's not being recombined and created through crossing over in meiosis. We've got capital R and capital Y. But these 2 options here, capital or dominant R and recessive Y, and this one is the dominant Y, recessive R. That is our new combinations of alleles in the gametes, and it's created these 2 new gametes. But crossing over is rare. So that means you won't get very many of these 2 gametes, and you'll get lots of this gamete, lots of this gamete. And that then leads into our answer. So explaining these results. Yellow and round, the top one, these are the 2 genotypes that you could get from the non-crossing over possible gametes. And you could also get the green and wrinkled from 2 lots of the lowercase r, lowercase y joining together. In fact, that's actually written here as well. So those are the most common, the top and the bottom are the most common because those gametes are not a result of crossing over in meiosis, and crossing over is rare. Now, the reason we have fewer green and wrinkled compared to yellow and round is there's 2 possible genotypes compared to only 1 for those phenotypes. And that's because green and wrinkled is recessive. So here we can see the heterozygous genotype still gives us yellow and round. So that's why there's the most yellow and round, then the green and wrinkled. The 2 phenotypes in the middle of the table are not as common, and that's because crossing over is rare. So we don't make very many of these 2 gametes. And that then means we don't get very many of these genotypes, which code for those phenotypes. So the key thing with a question like this is pointing out autosomal linkage occurred, if they haven't already told you, and also pointing out crossing over has occurred. But that's rare, which is why we don't get very many of these gametes and therefore those phenotypes. So sometimes the mark scheme does insist that you literally pick out which gametes were a result of crossing over, which were not, and link that to the numbers of each phenotype. So you could be asked to do a statistic on this, and it would be chi-squared, because you're looking at how many individuals fit into a category, or in this case for inheritance, how many individuals have a particular phenotype. And if we want to see, is there a difference between what we expect based on our Punnett square and what we actually observed, we can do a statistic to see if it's a significant difference between what we expected and what we observed. So here's an example. Going back to what we did at the start, cystic fibrosis caused by a recessive allele. We said we expected there to be 12.5% probability of it being a girl with cystic fibrosis. We could then actually look in the offspring and count what proportion did have cystic fibrosis and were a girl, and see what we expected. Is that significantly different to what we observed? Now, you wouldn't usually do that with humans because there's not going to be a large enough sample of the offspring from those 2 parents for it to work. But we've got an example here with corn, looking at the colour of the corn kernels. And in this example, we have got purple and yellow corn. Purple is dominant, yellow is recessive. Based on this Punnett square, we expect a 3:1 ratio of purple corn kernels to the yellow colour. So that's what we expect. We're then told that a student counted the actual number, so this is what they observed, and they got 21 purple kernels, 13 yellow. And we have to say, does that follow what we expected? So our null hypothesis would be there is no significant difference between the expected and observed frequency of the colour of corn kernels. And then we can fill in a table like this. Now, you wouldn't be expected to remember how to draw this table, but it has come up that they've given you this table with empty sections and you then have to fill it in. So you need to know that O is the observed, E is the expected. So here's what the student observed: 21 purple, 13 yellow, which means there were 34 in total. We said we expect a 3-to-1 ratio, but we need to turn that ratio into frequency. So that means we need to know what 75% of 21 is and 25% of 13. And I've calculated that here. O minus E is your observed frequency minus your expected frequency. So we've got that result here. Then we square that. So even if it was negative, because it's squared, it all becomes positive. And then we divide that by the expected frequency. The sum of this column is your chi-squared value. So we've got a chi-squared value of 3.176. You could be asked to work out the degrees of freedom, and that is the number of categories minus 1. We've got 2 categories, purple or yellow, so we have 1 degree of freedom. Now, if we were to look at this in a critical values table, it's a bit blurry, but we need to look at the P equals 0.05. That means that we'd have less than 5% probability that the difference between observed and expected is due to chance. We've got 1 degree of freedom. We've picked out the column that has 0.05. So we are comparing to this critical value, 3.841. And the critical value is like a threshold. You've got to see, does your chi-squared value meet or exceed that critical value threshold? And it has to meet or exceed it to be able to say there is a significant difference. So ours doesn't. Our chi-squared value is lower than the critical value. And there'd be a mark for stating that, which I've put here. So what that means is there is more than 5% probability that the difference is due to chance, not R, I should say is. And you do have to state difference and not just result to get the mark. So that means we can accept the null hypothesis, and there is no significant difference between what we observed and the ratio that we expected. Or in other words, the corn kernels did show a 3-to-1 ratio because it wasn't different to what we expected, and we expected 3-to-1. Next then, 3.7.2, populations. And this is where the Hardy-Weinberg principle comes in. It's normally assessed within the inheritance questions though as well. And Hardy-Weinberg is a mathematical model that can be used to predict allele frequencies within a population. So here are just some definitions to check. Turn them into flashcards if you want, because they are commonly assessed. Gene pool, that is all the alleles of all the genes within a population at one time. Population, this comes up as a 2-mark question now. All the individuals of one species in one area at one time. 2nd mark, that can interbreed to make fertile offspring. Allele frequency, the proportion of alleles within the gene pool. So here's the Hardy-Weinberg equation. And in fact, although the top one is the equation, you have to use the top and this one here simultaneously to get your answers. So you need to know that lowercase p, that is the frequency of a dominant allele in the population. Q is the frequency of the recessive allele. So a single letter by itself is representing the frequency of an allele. P is dominant, Q is recessive. And if you add all of those together, that equals 1 because that's all of the alleles for that gene in a population. P squared is the frequency of genotypes. Specifically, this one is the dominant genotype. 2pq is the heterozygous genotype frequency. Q squared is the frequency of the homozygous recessive genotype. And again, if you add all of the genotypes together, it equals 1 because those are all of the possible genotypes you can have for this particular gene. So in this question, cystic fibrosis is caused by a recessive allele. 0.02% of the UK population suffer from cystic fibrosis. What proportion of the UK are carriers? So step 1 is always identify from the 2 equations, what information have you actually been told? So we have been told that 0.02% suffer from cystic fibrosis. And cystic fibrosis is caused by a recessive allele. So to have cystic fibrosis, you have to be homozygous recessive. So 0.02% is Q squared. But we can't have it as a percentage, so I've converted it. Then we need to work out what we need to identify. And we've been told what proportion of the UK are carriers. Carriers, that would be heterozygous, so they want us to work out 2pq. And the strategy for Hardy-Weinberg is always work out which bit of the formula you have, then which bit you're trying to identify, and to do this, you then always need to work out what p and q are. So q, we know what q squared is, so if we square root that, we then get what q is. And now we've got q, we can use this formula to work out p. Rearrange it, 1 minus 0.014, this is what p is. So now we are working out what proportion are heterozygous. So for heterozygous, it's 2pq, so we then just need to do 2 times p times q. And that comes to 0.03. And they've just said proportion, so we can leave it as that, or you could write 3%, but do look carefully to see if they've asked for your answer as a percentage or just proportion. Next then, 3.7.3, evolution may lead to speciation. So variation, individuals within a population of a species may show a wide range of variation in the phenotype, and that is because of both genetic and environmental factors. If it's genetic variation, the primary source of genetic variation is mutations, but also genetic variation is introduced through meiosis and the random fertilization of gametes. Predation, disease, competition for the means of survival. Basically, this means there's going to be competition within populations. There's going to be disease, which means some individuals are more likely to survive, and there might be predators, and therefore if you're more likely to evade a predator, you're more likely to survive. And this then leads to natural selection. And the organisms with the phenotypes providing a selective advantage, meaning they're more likely to survive, they are also then more likely to reproduce, pass on those beneficial alleles to the next generation, and that's how we get this differential reproductive success, or in other words, the allele frequency for that gene might change in the gene pool. Now, all of this is in topic 4 as well as topic 7. And I go through that in more detail in the entire topic 4 video, because this is just a brief summary that they do in topic 7. And basically they do it so that you can be assessed on this content on paper 1 and paper 2 and paper 3. So types of selection. There are 3 different types. Stabilizing selection. So by type of selection, we mean the outcome of natural selection. Here's our original population looking at number of individuals for each trait. And the modal trait is the most common, or the middling feature, so medium color is the most common. The very pale, the very dark are not as common. And stabilizing selection does favour the average phenotype. And individuals with the extremes are less likely to survive, therefore less likely to reproduce, and therefore over many generations you lose those extreme traits, and the middling trait that was already the most common becomes even more common. And the example that you learn in topic 4 for this is human birth weight. Directional selection also came up in topic 4. This is when one of the extremes provides selective advantage and is favoured in those environmental conditions. So maybe there's an environmental change, which now means this extreme trait provides the selective advantage. So over many generations, more individuals now have the extreme trait, the middling trait's less common, and the other extreme is almost completely lost. And the example that you learned in topic 4 was antibiotic resistance, because those with the most resistance were more likely to survive. The new one for year 13 is disruptive selection. And this is when both extremes are favoured, meaning they both provide a survival advantage. So if an individual has either of those extreme traits and allele, they're more likely to survive, reproduce and pass on that allele. And this can then lead to the process of speciation. Now, you don't have to know an exact example for this like you did for the other 2, but an example could be maybe birds with very small or very large beaks are more able to survive, because birds with very small beaks might be able to get their beaks into tiny holes in trees to eat insects, and the birds with bigger beaks are able to crack seeds and nuts and eat that. But if you've got a medium-sized beak, you're not very good at either. So you're not going to get as much food. So this then takes us into the concept of evolution, genetic drift, and speciation. Evolution is a change in allele frequency within a population over many generations. And that can also lead to speciation. Speciation is the process that results in the creation of a new species. And this is going to occur when one original population of the same species become reproductively isolated. And this isolation means you now have two separate populations of the same species, but they do not breed to make fertile offspring at this point. And that's what reproductively isolated means. It means they cannot reproduce. Now, the reason that might happen is different, and we're going to come to that. But the outcome, if you've got these two separated populations that are no longer reproducing, both populations will have random mutations happening. But because they're no longer reproducing together, those mutations stay within this population. Mutations that anyone has in this population stay there as well. And that means over time, you get this accumulation of differences in the gene pools, so that if they were able to start reproducing together again, they'd be so genetically different that they cannot interbreed to make fertile offspring, and therefore they're now classed as a different species. So that is our process of speciation. And what causes them to be reproductively isolated is either a geographical barrier, which is known as allopatric speciation, or they might be in the same location, but they still don't reproduce. And that's known as sympatric. So allopatric speciation is when they're reproductively isolated because of some geographical barrier. And that could be a new mountain range, a body of water, but they just can't reach each other. All of the other marks would be exactly the same. So you always have that concept of they are reproductively isolated. You have to state whether that is geographical, allopatric, or sympatric, which we'll get to. That means that different mutations will accumulate in both populations, and therefore they'll go through natural selection and evolution for their environment. You get an accumulation of different mutations, and therefore they're now so genetically different they cannot interbreed to make fertile offspring. So they're two new species. And this is an example of directional selection. Sympatric, this is when populations become reproductively isolated, not because of a geographical barrier, but due to something else. And it could be their behaviour. So it could be that linked to the idea of courtship, or it could be as well that they are now producing gametes at different times of year. So they're fertile at different times of the year. So even if they are mating, they're not making offspring because one of the pair in that reproduction is not making a gamete. And then you get the same marks again. So for genetic drift, this is the change in the allele frequency within a population between generations. And there will always be genetic drift from one generation to the next, but continual, substantial change or genetic drift is when it's classed as evolution. And in smaller populations, genetic drift has a bigger impact because if you have a slight change in the number of alleles, in a small population, that's a big change in terms of the frequency. Because if you just change 2 out of 10, that's 20% change. If you change 2 out of 100, that's only 2% change. So the smaller the population, the bigger the impact. And that's why smaller populations evolve more rapidly. Next then, 3.7.4, populations in ecosystems. And again, lots of key terms here. Population is a group of organisms of the same species living in the same area at the same time, and they can interbreed to make fertile offspring. Habitat is the part of an ecosystem in which a particular organism lives. Community is all the populations of different species in the same area at the same time. Ecosystem is a community and the non-living components of the environment. This bit here isn't part of the definition, but just being aware that ecosystems vary in size. Niche is an organism's role within an ecosystem, and that includes its position in the food web and its habitat. And this is going to be determined by adaptations to both the abiotic and biotic factors they're exposed to. Carrying capacity is the maximum population size an ecosystem can support. Abiotic factors, those are the non-living conditions of an ecosystem. Biotic factors is the impact of the interactions between organisms. So a bit more on these two, abiotic and biotic factors. We said that the population size and the carrying capacity are dependent on abiotic and biotic factors. Abiotic factors are the non-living factors, so that could be things like temperature, oxygen concentration, carbon dioxide concentration, light intensity, pH, soil conditions, and the key kinds of questions you get are either linking to something else in the specification to explain why any of those would affect plant survival or animal survival. And that might link to photosynthesis, might link to enzymes. And then it's knowing maybe links to how they could evolve. So that would be the idea of natural selection. If they're better adapted to that environment, they're more likely to survive, pass on those alleles to the next generation. And then eventually, over many generations, much of that population will have adaptations to those abiotic factors. Biotic factors, that is looking at things like competition and predation. Competition can be either interspecific or intraspecific. Interspecific competition is when members of different species are competing. And the way to remember this, I think, is international travel, spelt inter, T-E-R, is when you're traveling to another country. So it's a different country. So interpacific competition is a different species. Intra is when members of the same species are competing. They'll compete for the same things. It's just if it's members of the same species, they're also competing for a mate. And this is for animals and plants. It's not just animals that compete. Plants would also be competing for water availability in the soil, carbon dioxide concentration, mineral ion concentration in the soil too. Predator-prey relationship, so this is still biotic factors. Looking at the interaction between the predators and prey in a food web, you get this typical shape graph, regardless of the species, where we've got the predator shown in blue and the prey shown in red. And you always see these three features. The size of the predator population is usually going to be lower than the size of the prey population. Both populations will fluctuate in size, and there's a bit of a lag time. You'll see a change in the prey before you see the change in the predator population. And this is because the predators are eating the prey. So there has to be more prey than there are predators to sustain them. They both fluctuate because as the predators are eating lots of prey, that causes their population size to decrease. That will then cause the predator population size to decrease. But because the predators have gone down, the prey can then increase, and this happens over and over. So the last little bit then is looking at sampling, so that you can look at population sizes. And the reason we sample is it's going to be more accurate and less time consuming than trying to count every single individual. But we have to make sure that the samples we take do accurately represent the population. So that would mean you have to do random sampling to avoid bias. And that's if it is a uniform area, meaning it's equal distribution. If it's not equally distributed, you do systematic sampling, which is a line transect or belt transect, to examine that change over distance. And then also it has to be a large number of samples to ensure your sample is big enough to accurately represent the population. Now, if you say large number, that should be fine. If you're going to state a number, I would say 30 plus, just to show it really is large. Now, the different ways that you can sample then. First thing is thinking about what type of organism you're estimating the population size of. So if it's slow moving or it doesn't move, like a plant, then you'd use a quadrat. If it's a motile, meaning moving organism, like a bird, you would use mark, release, recapture. If you've got uniform distribution, you do random sampling. If it's uneven distribution, it's systematic sampling, which is your belt transect or line transect. We're going to have a look at that first, and then we'll come back to the mark, release, recapture. So random sampling. First of all, you would lie 2 tape measures at right angles to create a gridded area. Then use a random number generator to generate your coordinates. That then tells you where to place your quadrats. You go along those 2 numbers of your coordinates. Depending on what the question says, will determine how you're estimating in your quadrats. Because it might say you need to do percentage cover, in which case you're estimating inside of your quadrat what percentage of it is covered by your plant. Or it might ask you to estimate the number, and in which case you do density, which means count the individual number of plants that there are there. Frequency doesn't tend to come up, but local frequency is the idea of you would use a gridded quadrat and just count how many of the squares it's present in. So it's slightly different to percentage cover, because that you're estimating the percentage covered. The local frequency is just, is it present or not in the tiny squares? Now you have to make sure it's representative. So you have to do this a large number of times, so at least 30 times, calculate a mean. And then if it's percentage cover, it ends there. If it's density though, meaning you counted the number of plants in the quadrats, you now have your mean number of plants in a quadrat, but we want to know it for the entire field. So there'd then be a mark for describing how you'd work that out. So you'd need to know what the area of the field is and the area of a quadrat to see how many quadrats would fit into that field. And then whatever your mean is, you're multiplying that by the number of quadrats that fit into the field. Another thing that they've become really picky on is if they've said describe how to work out the mean number, you also have to actually describe how to calculate the mean and not just say calculate it. So you'd need to say add up all of your 30 repeats and divide by 30. A line transect, this is used to estimate population size when there are that uneven distribution of species. So that could be things like on a rocky shore, if you've got a river in the middle of a field, and you'd place a tape measure to act as your line transect, going through all of those different sections so that you then do get information for all of the different parts. And a belt transect means that you've got your tape measure and you're lining up quadrats next to it. An interrupted belt transect is what is most commonly used. This means you place your quadrat at intervals along your tape measure. And it might be every 5 meters, every 10 meters. However far you start to notice a difference, that's the frequency that you'd need to place them to make sure you don't miss any important information. You still have to repeat multiple times though. So here would be your method if you were asked to describe a line transect. Method, that is. So place your tape measure at a right angle to the shoreline. So that would mean it's going through all of those different sections that we want to see. Place your quadrat every 5 meters. It doesn't have to be 5, but it's at stated intervals. Collect your data, just like we said last time, so either density or percentage cover. And repeat again a large number of times, calculate a mean. But for this one, the repeats would be multiple repeats at parallel, so in parallel to your original one, along the beach or whatever area it is that you're measuring. So then we've got mark, release, recapture for if you are trying to estimate the population size of a motile organism. Here is our method. So you need to capture an initial sample. You need to mark them. So that might be putting a bit of paint on a snail shell, for example. Then you release them back into the wild. You then leave them for a period of time after they've been released so that they can randomly disperse throughout the habitat. Then you take a second sample, and the total number captured in the second sample and the number recaptured, meaning the ones that have the mark on them, are recorded. Then we can use the formula to calculate our estimate. So to work this out, we'd need to do the number of organisms initially caught in that first sample, times by the number of organisms in the second sample, divided by the ones that had the mark on them. So that would then give us, in this example, we've got 20 butterflies were initially caught. In the second sample, there were 22 butterflies, 9 of which had the mark. So we do 20 times 22 divided by 9. That comes to 48.89, but we need to have whole numbers here because it's numbers of organisms. So I'm going to round up to 49 butterflies. There's always a mark for stating this formula when you have a method question for mark, release, recapture. So if in doubt, always state that formula. There's questions linked to the ethics of this sampling as well. So you need to consider that when you're marking the initial sample, that the way you mark them is not with a toxic chemical. So if you are putting on paint, it has to be non-toxic. It mustn't increase their chance of being seen by predators and therefore eaten and killed. And it mustn't reduce their chances of reproduction. So those are the ethics. Now, a non-ethical consideration of the mark is it needs to be weather resistant, because you don't want it to be something that's going to wash off when it rains, because then they're not going to be marked anymore. There's also assumptions linked to this. The estimate might not be accurate because the population size isn't actually constant, and we are assuming it is. And for a population to be constant, that means you have no births, no deaths, no migration. And that doesn't happen in any population. So we're assuming the population size is constant. In reality, it's not. So that's one reason why our estimate will be inaccurate. And also, we're assuming that the animals, when we release them and leave them to evenly distribute, we're assuming that they are going to evenly distribute. But in reality, they don't often do that, because lots of animals will probably huddle together near a water source or a food source, or where they feel safest. Then we go on to succession, and this is the change in ecological community over time. A primary succession starts with a pioneer species, and that is going to colonise. It might be bare rock or sand. You don't have any soil yet. You don't have to know any examples, but just to give you a bit of information, pioneer species could be things like lichen. Lichen are a mixture of fungi and algae, and they can survive in really harsh abiotic factors. And through the death and decomposition of the lichen, or whatever the pioneer species is, that is going to change the environmental factors, making them less harsh, less hostile, and you get this thin layer of soil, and then larger plants are able to survive there. And that might be things like mosses or smaller plants. And they will further increase the depth of the soil and the nutrient content, because they'll have roots that will start to hold the soil together. And as they die and decompose, they're going to add more nutrients to the soil. And this pattern continues over and over. Each new species is going to improve the environment so it's less hostile. But that then does mean that bigger species, plant species, can arrive and they'll outcompete the previous one. So the changes that organisms produce in the abiotic environment can result in a less hostile environment and therefore increases the biodiversity. And the final stage of a succession is known as the climax community, and this is dominated by trees. Now, to give you the marking points, because that was more a full explanation, the actual marking points on a long answer question are very brief. There's always a mark for saying it's initially colonised by a pioneer species. Next, it's saying the pioneer species changed the environment. The environment becomes less hostile for new species. There's an increase in biodiversity, and it finishes with a climax community. That would get you full marks on a 5 marker for describing succession. So screenshot that or turn it into a flashcard to learn those key points. So here's our summary from succession. The species richness increases, the number of organisms increases. That's why the biodiversity is higher. So as succession occurs, we get larger plants. That is going to attract animals as well, and you end up with these more complex food webs. Now, you can prevent succession from progressing, and that is a technique to conserve habitats. And this is because as a succession moves through its stages, you ultimately get a climax community where you have lots of large trees. And those large trees will outcompete the smaller plants because they're going to be shaded. So if you just let succession occur, all of the area would be forest. And that's what it used to be in England, for example. It used to just be forest everywhere in the UK, not just in England. But then we cut down the trees, started to build. But you can manage forest as a way to maintain maybe one of the earliest stages of succession. to have one section of the forest thriving with grasses, another section of the forest thriving with smaller plants, and then some of the sections thriving with trees. And the idea here is you get a greater variety of habitats. That then means you've got a greater variety of food sources, and a greater range of animal species will be able to survive there, and also plant species. So you might do things like coppicing or pollarding, where you cut down some of the trees, but you don't take out the roots, because then over time the trees can still regrow, so it's not going to be harmful for the environment. And also it means you get timber for furniture, or you could use the wood for fuel as well. Topic 8 for AQA A-Level Biology, and we begin with looking at 3.8.1, alterations of the sequence of bases in a DNA can only alter the structure of proteins. So basically it's mutations, and a gene mutation is the alteration of a base in the sequence of bases for one gene. And this is probably going to occur during DNA replication, which is in S phase of interphase in the cell cycle. Gene mutations occur spontaneously, but the frequency at which they occur can be increased by exposure to mutagenic agents. And that's things like carcinogens and UV light or ionizing radiation. And because a mutation can change the DNA base sequence in a gene, that means it could result in a different sequence of amino acids in a polypeptide. And if that sequence of amino acid changes, then that can alter where the tertiary structure bonds will form, like the hydrogen ionic bonds, and that could result in it folding into a different 3D shape. And if a protein has a different 3D shape, it will have a different function and potentially make it a non-functioning protein. Alterations to genes like this can result in a mutation that could cause cancer, and cancer comes up a little bit later in this topic. So there are 6 different types of gene mutations, 2 of which you learn in topic 4, but now you learn addition. You've already covered deletion and substitution in topic 4, inversion, duplication and translocation of bases. So addition mutations is when one extra base is added to the DNA base sequence. So if we have a look at this original sequence here, we've added in an additional adenine nucleotide, and as a result we get a frame shift, meaning all of the bases downstream of that mutation shift one position, and they all shift one position to the right. And that causes a change in all of these codons, and therefore they will probably code for different amino acids, and that will result in a very different amino acid sequence and a non-functioning protein. Deletion is when one of the bases in your DNA base sequence is deleted. This also causes a frame shift, but this time all of the bases downstream of the mutation shift one position to the left. Again, you get different codons, therefore it's going to code for different amino acids and a very different amino acid sequence, and therefore a non-functioning protein. Substitution is when only one base is swapped, so it's just changed. You still have the same number of bases, and it will only affect the one codon that base is in. And because the genetic code is degenerate, meaning you have multiple codons coding for the same amino acid, even though this mutation has happened, sometimes it still results in the codon coding for the same amino acid, and therefore has no overall impact on the final protein. And if the mutation happens in the introns within a gene, which are the non-coding regions, again, it's not going to change the amino acids that it codes for. So here you can see an example. We've just changed the thymine nucleotide into an adenine nucleotide. Inversion is when a section of bases will detach from the DNA sequence, but when they rejoin, they are inverted, meaning they're back to front now. So this section of the code is back to front, and you get a completely different amino acid sequence. So we can see our original DNA here, and now we've got this section must have broken off, and when it rejoined, it's reversed itself, it's inverted. So that is going to code for 2 completely different amino acids now. Duplication is when one particular base is duplicated at least once. And because that means we're adding in bases, it's going to cause a frame shift to the right downstream of that mutation. So you end up with a different sequence of amino acids. So here's an example here. We've got the base thymine, added twice. And then lastly, translocation is a section of bases on one chromosome detaching, and it attaches onto a different chromosome. This is a substantial alteration, and therefore it can cause significant impacts on gene expression, and therefore the phenotype could be very, very different. 3.8.2 then is gene expression. And we start with stem cells. And in multicellular organisms, there are a diverse range of specialised cells, but they all originated from undifferentiated stem cells. So a stem cell is an undifferentiated cell, meaning it hasn't become specialised yet, that can continually divide, and it can then go on to differentiate to become a specialised cell, as we can see here. And there's different types of stem cells based on their ability to differentiate. So we've got totipotent, pluripotent, multipotent, and unipotent. Totipotent stem cells can divide and produce any type of body cell. So these are able to differentiate into all cells. And they occur for a very, very limited time in early mammalian embryos, so shortly after the sperm and egg fertilise. Pluripotent are still found in embryos, so we can see here, you don't need to know this term blastocyst, but the embryo then develops into this blastocyst. The cells on the outside go to form the placenta, and the ones here in this inner mass are the pluripotent stem cells. So these pluripotent stem cells can differentiate into almost any type of cell, just not the placenta, because those are the ones on the outside that have already started to differentiate, but any other cell. And these are the ones that are typically used in research or to treat human disorders. So the stem cells could be used to regrow damaged cells in humans, such as to replace burnt skin, or even beta cells to try and treat type 1 diabetes, or neurons to treat Parkinson's disease. There are issues with this though, as sometimes the treatment doesn't work, or because stem cells can continually divide, they do continually divide and they can form tumours. There is also debate as to whether it is ethically moral, because in order to use it for treatment, you would have to make a therapeutic clone of the person you are trying to treat. And the embryo is then destroyed once the pluripotent stem cells are removed as well. So we also have multipotent and we have unipotent stem cells, which are found in mature mammals, meaning after the embryo stage. And they can divide to form a limited number of different cell types. Multipotent cells are, for example, bone marrow cells that we have as adults or even as children. When we say adults, we just mean you've been born in this instance. And they can differentiate into a limited number of cells, such as all of the blood cells. Whereas unipotent cells, uni meaning one, they can only differentiate into one type of cell. And an example of that is the cardiomyocytes. They are unipotent stem cells which regenerate into new heart muscle cells in small numbers. Now, induced pluripotent stem cells, this is something that has been produced scientifically from adult body cells. Somatic cells means body cells, using appropriate protein transcription factors to basically ensure that all genes can be transcribed again. And therefore, it makes it pluripotent in property, even though you might have taken a multipotent or a unipotent stem cell. And this overcomes some of the ethical issues because you're not then creating a clone embryo, doing therapeutic cloning, and you're then not destroying embryos. You're getting permission from an adult, taking some of their body cells and reverting them back into a pluripotent state. So the way this is done then, the iPS cells are created from the adult unipotent cells. These are altered in the lab to return them to that state of pluripotency. The genes that were switched off, which basically means those genes could not be transcribed, are now able to be transcribed again. And this is done using transcriptional factors. iPS cells are very similar to embryonic pluripotent stem cells, but as we said, it doesn't have those same ethical issues, such as the destruction of an embryo and not being able to get permission. have shown a self-renewal property in that they can divide indefinitely to give limitless supplies. And for these reasons, they could be used in medical treatment instead of embryonic stem cells. So if we have a look, then we've talked about this use of transcriptional factors in induced pluripotent stem cells, but we haven't gone through what that means. So you can control transcription. You can also control translation, but we're just going to focus on how you can control transcription first, meaning you can prevent it from happening or initiate it to happen. And this is actually what happens naturally when cells differentiate and become specialized, because every cell in the body has your entire genome, so all of the genes. But because of transcriptional factors and other things, but mainly transcriptional factors, some of those genes don't get transcribed, some of them do. And that's how you get cells that produce a particular range of proteins and become specialized. So in eukaryotes, transcription of target genes can be stimulated or inhibited using transcriptional factors. And that's what we're going to have a look at. So transcription of a gene will only occur when a molecule from the cytoplasm enters the nucleus and it binds specifically to the promoter region on DNA once it's in the nucleus. And those molecules that do that are proteins, and they're called transcriptional factors. Each one can bind to a different and particular base sequence of DNA. And this links back to topic 1, knowing that because transcriptional factors are proteins, that means they have a unique tertiary structure. So it gives it that unique 3D shape. And that means it will be able to bind to a sequence of bases complementary in shape. So remember that concept of tertiary structures and complementary shapes. So once it does bind, it can then stimulate RNA polymerase so that transcription begins. And that is how it's going to be initiating transcription. So we do then get an mRNA molecule that can then go on to be used in translation. So without the binding of that transcription factor, the gene is technically inactive because transcription is not happening, and therefore that protein isn't being made. And an applied example that you need to know about is estrogen. Now, estrogen is not the transcriptional factor, but it activates the transcriptional factor. Estrogen is a lipid-soluble hormone because it's a steroid hormone, and steroids are a type of lipid. So that lipid-soluble hormone, which is estrogen, can dissolve and move by simple diffusion from outside of a cell into the cytoplasm. And when it's in the cytoplasm, there's a transcription factor that has a receptor on it, which is complementary in shape to estrogen so that they can bind. And when it does bind, it causes a change in the tertiary structure of that transcription factor. And that change in the tertiary structure changes the DNA binding site so that it is now complementary in shape to a particular sequence of DNA bases. So that activated transcriptional factor moves into the nucleus. It will then bind to the promoter region of a target gene, which has the specific DNA base sequence that it's complementary to. And when it does that, it stimulates the RNA polymerase to bind and transcription can occur. And then we get our mRNA molecule that can be used in translation. There's also another way that transcription can be controlled, and that is through epigenetics. Epigenetics, here is your definition, is the heritable change in gene function, but without changing the DNA base sequence. So 2 marks there for those 2 parts of the sentence. And these changes are caused by changes in the environment. So for example, stress and diet. And it can result in transcription being inhibited or initiated. And that is due to particular chemical tags that bind either to the DNA or to these histone proteins. So one of those chemicals is methyl groups. And methylation of DNA means methyl groups binding to DNA, which we can see here. These green circles are representing methyl groups binding to the DNA. And when those methyl groups are added to DNA, they attach to cytosine bases only. And this prevents transcription factors from being able to bind. And that is because when the methyl groups bind, it's going to attract the DNA and histone complex really tightly together. So it winds up like this. And as a result, 'cause it's so tightly coiled, you've got genes which are tightly wound up, and therefore transcriptional factors can't reach them to bind. That means RNA polymerase can't be stimulated and transcription doesn't happen. The next chemical group is the acetyl groups. And if you have acetylation of histone proteins, that means the addition of acetyl groups, and they can bind to the histone proteins. Now, an acetyl group has a partial negative charge because it's got an oxygen in it. And the DNA also has this partial negative charge because it's got a phosphate group in it. And those two negatives will repel. And that's why the DNA and histone complex is not as tightly coiled. And as a result, the DNA is accessible. Transcriptional factors can bind to the promoter region on the DNA and stimulate RNA polymerase. So that means if you have decreased acetylation, meaning the acetyl groups are being removed from the histone proteins, that makes the histones more positive, and therefore they're going to attract to the DNA, and your DNA and histones are going to be more strongly associated, and it coils back up again. And again, that means that transcription factors can't bind. So that means that if you have increased methylation and decreased acetylation, that results in this tightly coiled complex of the DNA and histone proteins, and that will inhibit transcription. Whereas if you have methyl groups removed, so decreased methylation or increased acetylation, then you get this complex where it is not as tightly coiled and the genes might be exposed so that transcriptional factors can bind, and that initiates transcription. Now, you can also have control of translation after transcription has happened and mRNA has been made. And that is what RNA interference is, or RNAi for short. In eukaryotes and some prokaryotes, translation of the mRNA produced from the target genes can also be inhibited because of the RNA interference. So the mRNA molecule has already been transcribed, but that mRNA molecule gets destroyed. It basically gets cut up into small pieces, so it can't then attach to the ribosome and be used in translation. And small double-stranded RNA molecules are involved in this, typically known as siRNA, so small interfering RNA, or miRNA, microRNA. So here's our flow diagram of how protein synthesis typically happens. We've got transcription, that results in the production of mRNA. mRNA is then translated, and that results in the production of a polypeptide chain. So that is how protein synthesis occurs. Now, we've already talked about transcription could be prevented because, or initiated because of transcriptional factors and epigenetics. But here is where our siRNA or RNA interference has a role. So siRNA can cut up the mRNA to prevent translation, and therefore small interfering RNA results in RNA interference. So RNAi, the reason this is important is it's regulating gene expression, controlling development, and also it can fight viral infections and potential therapeutic uses for this could be silencing harmful genes in disease. So that's when it's going to be naturally occurring, and it could also be used in medicine in this way. The way this works then is, in terms of siRNA, which we can see here, siRNA is produced from double-stranded RNA, and it splits into a single strand. It then is going to bind to a protein to make a complex, and one strand of that siRNA will bind to a complementary sequence of mRNA that's just been transcribed. And once it's bound to the siRNA in that protein complex, that protein is actually an enzyme that can cut up the mRNA. That is the level of detail that you need to know. I know it might sound a little bit vague, and that's because it is, but that's the extent that you need to know. It quite often comes up as application questions where they then give you a little bit more information. An alternative is miRNA, which was the microRNA, and miRNA is not fully complementary to its target mRNA molecule. So instead it binds to multiple mRNA molecules, And that often causes the blocking so that the mRNA can't attach to the ribosome, or it can also cause mRNA degradation as well. But either way, it results in translation not happening. So this then follows on to cancer. And cancer can result from mutations in genes that are responsible for controlling and regulating mitosis, as well as we're going to have a look at the role of epigenetics as well. But first of all, if these genes do get a mutation and a non-functioning protein is made, then that means mitosis is not regulated and you get uncontrolled mitosis or cell division, and that results in the creation of a tumour. Not all tumours are cancerous. They can be benign or malignant. So benign tumours, these are not cancerous. They grow, or they can grow very large, but just at much, much slower rates. And they're non-cancerous because they produce this adhesive molecule which sticks all of those cells together in a particular tissue, and they're often surrounded by a capsule. So that means that they're much easier to remove by surgery because you can remove the entire tumour, all the cells, because they're encapsulated. And also because it's in that capsule, it's localised, it's not going to break off and spread to other tissues. So it's often not life-threatening. However, that does depend on the location of the tumour because it will continue to grow and the treatment is surgery to remove it. So if it's in a location of the body where surgery would be difficult to remove it, such as in the brain, it could still be life-threatening. Malignant tumours are cancerous and they can grow large and they can grow large very rapidly. The cell nucleus becomes large and the cell can then become unspecialised again. They don't produce that sticky adhesive layer and they also don't have a capsule around them, which means that these tumours are able to metastasise. And what that means is cells from the tumour can break off and get into the bloodstream or the lymphatic system and therefore move around the body and then lodge in new tissues to make secondary tumours. The tumour, as I said, isn't encapsulated, and that also means it can grow out projections and reach blood supply. And as soon as it gets a blood supply, it's got lots of glucose and oxygen for respiration, which is why it can divide so rapidly. It can be life-threatening, and as well as trying to remove tumours, you can also have treatments such as radiotherapy and chemotherapy. But it's much harder to successfully remove the tumours because they're not encapsulated. It's very hard to tell if every single cancerous cell has been removed in surgery. And that's why it's often, if you do have surgery, accompanied with radiotherapy and chemotherapy to potentially kill any cells that might have been left behind in the surgery. So tumour development can be due to gene mutation in either a tumour suppressor gene or an oncogene, which are two genes involved in controlling mitosis. Or it could also be linked to epigenetics if there's abnormal methylation of a tumour suppressor gene or oncogene, or increased estrogen concentrations linked to the idea of transcriptional factors. So let's just go through what these two genes are. Oncogenes are the mutated version of a proto-oncogene. And a proto-oncogene creates a protein involved in initiating DNA replication and mitosis when the body does need new cells. An oncogene mutation results in that gene permanently being activated. So you are going to have the protein constantly being produced to initiate DNA replication for cell division, even if new cells aren't being made, and that's why a tumour forms. Tumour suppressor genes produce proteins or code for proteins that slow down cell division and cause cell death. So when you no longer need new cells, it makes the protein, and also cell death if a copying error is detected, so that cell needs to be destroyed. Now, if a mutation results in the tumour suppressor gene, that means you won't make that protein anymore, and therefore cell division isn't going to slow down. And also potential mutated cells aren't identified and destroyed. And again, that is why it can result in a tumour forming. So if you have abnormal methylation, which links back to epigenetics, that means that this methylation could result in a gene being inactivated. And if that's the tumour suppressor gene, becoming hypermethylated, meaning lots of methyl groups are binding to that DNA. That's going to cause it to tightly coil. The transcriptional factor can't reach the gene, the tumor suppressor gene, and the tumor suppressor gene is not transcribed. So it means you don't get that protein that's needed to control the cell cycle. Or the opposite could occur in an oncogene, as they might be hypomethylated, meaning you don't have enough methyl groups binding, and as a result, the gene is permanently being transcribed. Now, the link to estrogen is after menopause in females, estrogen is no longer produced by the ovaries. While the menstrual cycle is active before menopause, the ovaries is the main place that estrogen is produced. But after the menopause, the ovaries stop making estrogen, and instead it's mainly produced by fat cells in the breast tissue. So in postmenopausal women, this local estrogen production, constantly being exposed to the breast cells, increases the risk of breast cancer. And that goes back to what we looked at in transcriptional factors, because estrogen can bind to receptor proteins on transcriptional factors to activate the transcriptional factor. And that transcriptional factor can then bind and initiate transcription. So if estrogen stimulates transcription of a proto-oncogene, it can cause that gene to be constantly transcribed, and therefore you're going to be making that protein all the time that stimulates cell division, and that can contribute to a tumour forming in the breast tissue, because that is where this estrogen is being produced. And this can result in a positive feedback loop, because if you're making more tumour cells, which are breast tissue cells that are making estrogen, your concentration of estrogen is now even higher in the breast tissue, so this process is going to happen even more. So that is why it can then happen very, very rapidly. Next, we move on to 3.8.3, using genome projects. So genome sequencing involves working out the entire base sequence of an organism's DNA. And projects have successfully sequenced the genome of many organisms, including humans. So this is just showing us the progress through that. It's a bit blurry, but many organisms have had theirs sequenced, and for humans it was finally completed, the Human Genome Project, in 2003, but it had been ongoing for many, many years. Scientists across the world all contributing to sequence our DNA. So the applications of this, first of all, in simpler organisms, for example bacteria, genome sequencing is very useful because it can be used directly to predict the proteome, which is all the proteins an organism can make in their cell. And that's possible because simple organisms like bacteria don't have non-coding DNA. They don't have any introns, they don't have other non-coding sequences, and also they don't have regulatory genes as well. So that means the relationship between the genome and the proteome is very straightforward, and it's very easy to predict all the proteins they'll potentially make. And this has been useful in identifying potential antigens that bacteria could make, and therefore making vaccines against those particular bacteria. In complex organisms, for example humans, predicting the proteome after working out our genome is more difficult, and that is because we've got large amounts of non-coding DNA, and also we have regulatory genes that affect gene expression. So that means that knowing the genome sequence doesn't always directly reveal which proteins will be produced. Now, there are constant advances in sequencing technologies, which is why the spec doesn't get you to learn any, because they probably become outdated within a year. So that's why instead you just need to have this awareness that sequencing methods have become increasingly rapid, far cheaper and cost effective, and automated. And because of that, large-scale genome projects have enabled researchers to sequence even more organisms in less time with greater accuracy. So we've gained even more information. We then move on to 3.8.4, gene technologies, and I mean the title goes on, but it's basically what we all call gene technologies. So gene technologies involves recombinant DNA. And recombinant DNA means you're transferring fragments of DNA from one organism or one species into another organism's DNA or another species' DNA. The organism that receives the DNA is called a transgenic organism or genetically modified. The reason it's possible links back to topic 4. The genetic code is universal, which means the same codons on mRNA will code for the same amino acids in all organisms. And that's why you can take the DNA from one species, put it into the DNA of a different species, and it will still code for exactly the same sequence of amino acids and make that protein. Now, as well as that, it also works because transcription and translation are universal as well. So that means genes from one species can be expressed in another. And that's because once you insert that DNA into the new species DNA, transcription will occur to make mRNA, and that mRNA will then be translated to make the functional protein in the host cell. So the ways that this has been used is producing insulin, for example, taking the human gene for insulin, putting it into bacterial DNA, and then bacteria will make the human protein for insulin. And that has been massively beneficial because bacteria can create large quantities very rapidly of insulin, which can be used to help treat diabetes. It's also useful to make transgenic crops with beneficial traits, for example, inserting genes so that they're naturally pest resistant, or even inserting gene function for research models to try and work out the role of genes in terms of helping with medical research. So how this is done then, I've split the rest of this topic into this flow diagram because from my experience teaching this, one of the challenges with the gene technologies topic is students learn all of the technology separately and don't see how it all links together, and therefore struggle when it gets to the challenging application questions when it is all linked together. So here it is and how it all links. One branch is genetic screening, and this is where you might screen to see if you have a particular gene. That leads to genetic counselling. Now this is done using gel electrophoresis, and it can result in personalised medicine. Genetic fingerprinting, that is how you can examine an individual's DNA based on the VNTRs, those variable number tandem repeats. And that is used in forensic science, medical diagnosis, paternity tests and more. And one of the stages in genetic fingerprinting is gel electrophoresis. Then we have over here how we actually make recombinant DNA. And the first step is if we want to put the DNA from one organism into another, we need to get that piece of DNA to start with. And that's what creating DNA fragments means, getting the gene of interest that you want to insert into a different species DNA. So here are the processes of how you can do that. Once you've then isolated that gene of interest, we then need to get lots of copies of it before we go on to try and insert it. And you can either get large numbers of copies in vitro, which means outside of the body, and that is using PCR, polymerase chain reaction, or you can do in vivo cloning, which means making large numbers of copies of that gene inside of a cell. And that's typically done inside a bacteria, and here's all the processes how. So that's a very brief overview. Let's go into it in more detail, starting with how we create these DNA fragments. So the combining of different organisms' DNA, which could enable scientists to manipulate and alter genes to improve their industrial processes, or we looked at medical treatments, always starts with you have to get your gene of interest, which means isolating the DNA fragment. And there's 3 methods: reverse transcription, restriction endonucleases, and the gene machine. So reverse transcription, this is using an enzyme. And this enzyme makes DNA copies from mRNA. The enzyme actually isn't written yet, but it is reverse transcriptase. So that enzyme is an enzyme that can make DNA copies from mRNA. And it naturally occurs in viruses. So you learn in topic 2 how HIV has the enzyme reverse transcriptase, and that can make DNA copies from mRNA. So the reverse of transcription. A cell that naturally produces the protein of interest gets selected. Then large amounts of mRNA for that protein should be in that cell and they can be isolated. You would then mix that mRNA that you've isolated with the enzyme reverse transcriptase. And what that will do is reverse transcriptase can join DNA nucleotides together, so via a condensation reaction, creating phosphodiester bonds, based on mixing together DNA nucleotides, exposing it to this mRNA, and the complementary bases will be attracted and aligned, and then reverse transcriptase can join to create that new DNA molecule. But because we'll have one mRNA sequence, and then you get your DNA sequence aligning and being created against that, you end up making a single stranded DNA molecule. And we call this cDNA, complementary DNA, so complementary copy of our mRNA. So we then do have to make it double-stranded so that we can use it. And to make it double-stranded, we then need to make sure we've still got lots of DNA nucleotides present, and then we use the enzyme DNA polymerase. Now, the advantage of this method is that complementary DNA, or cDNA, won't contain any introns because mRNA doesn't have introns in it. They've already been spliced out. So that means we get a DNA copy with no introns in. And then we can see in this diagram, they then go to use PCR, so you get large numbers of copies of our cloned gene. Restriction endonucleases, this is another method using enzymes, and these are enzymes that can cut DNA. They naturally occur in bacteria, and bacteria have them as a defense mechanism. So if a virus was to inject their DNA into the bacteria, they naturally have restriction endonucleases to cut up viral DNA so that they can't be infected. And there are many different types of restriction endonucleases, and this links back to the idea of enzymes having a tertiary structure, which gives them a unique 3D shape, so their active site is complementary in shape to a particular sequence of DNA bases. So these restriction endonucleases, loads of different ones, so that they each have a different active site complementary to a different sequence of bases. They can then bind at that particular sequence, and they will break the phosphodiester bond at that point. Some enzymes cut at a location in a double strand to get a blunt end. What that means is it would cut directly down, so you get two straight lines of blunt end. But most of the time, and in terms of use, it's more useful than they would use enzymes that cut at a jagged line. And that then gives you an overhang of bases here and an overhang of bases there. And that is known as sticky ends. And sticky ends are useful because that means that they can align opposite another fragment with an overhang, and it's much easier then to join them together because a complementary bases can align. And this is going to happen at these palindromic sequences. And that means the bases, the order of the bases is the same forwards as the order of the bases is backwards. That's what we mean by palindromic. So where these enzymes can bind and cut is known as the restriction site or recognition sequence. Those are the two terms that we use to describe where the enzyme can bind. That is based on the active site being complementary to a particular sequence of bases, and it creates these sticky ends. A gene machine, this is when the DNA fragments can be created in a lab using a computerized machine. So first of all, the scientists will examine the protein of interest to identify what the amino acid sequence is for that protein. Then they can work backwards and work out what the mRNA sequence is, and therefore what the DNA sequence is. That DNA base sequence is then entered into the computer. Checks are made that you're not trying to make a protein that could potentially cause risk to human health, and other biosecurity checks, and that it's ethical to produce. Once those checks are in place, then the computer can create small sections of overlapping single strands of nucleotides, and these are called oligonucleotides. Those can then be joined to create the DNA for the entire gene. So once you've then created the entire gene, PCR is used to get large numbers of quantities and to make it double-stranded. So this process, the advantages are it's very quick, it's accurate, you can design it to be intron-free as well. And the reason we think intron-free is an advantage is prokaryotic cells don't naturally have introns. That means they don't have the cell machinery to splice introns out. So if we're going to be putting the gene into a bacteria, we need to remove the introns because they can't. So having it intron-free in the first place is a big advantage. Okay, so we've made our gene fragments. Now let's have a look at how you can clone them, meaning get large numbers of that gene of interest. First of all, with in vivo cloning. So we've already created the DNA fragment. Now we need to look at how we get that fragment into the vector and how we get that into the host cell. So first thing though is adding a promoter and terminator region. So when we insert a gene into a host cell, so basically when we take one gene and put it into the DNA of another organism, it's not enough to include just the DNA base sequence that codes for the amino acids. For transcription to successfully occur, you also have to add a promoter region at the start of the gene. And the DNA sequence is inserted before the gene. And that acts as a binding site so that RNA polymerase and transcriptional factors have something to bind to so they can initiate transcription. The correct promoter must be used for the host organism though, so that the gene is expressed properly. Now at the very end of the gene, a DNA base sequence is added as well, and that's called the terminator region. And that will signal the end of transcription and it ensures that RNA polymerase will detach and it stops transcribing at the correct point. So they are important then because without a promoter region, the gene wouldn't get transcribed. And without a terminator region, transcription might continue so that it then continues to transcribe unintended regions. And that would be wasteful of resources and also it might disrupt the other genes. So basically these sequences, the promoter and terminator region, are essential for being able to produce functional mRNA when you do this gene technology. So how we get that DNA into a vector, but first of all, what is a vector? So a vector is something that will carry the DNA fragment that we've made into the host cell. That's what a vector is. And the most common vector that we use is plasmids, so those small loops of DNA that some bacteria have. So that's what we're going to be doing, getting our DNA into the plasmid first of all. And the way that we do that is the plasmid has to be cut open using a restriction endonuclease, but you have to use the same restriction endonuclease as the one that you use to cut out your DNA fragment. And the reason for that is that means your plasmid and your DNA fragment will have the same sticky ends. So that means they'll have complementary sequences overhanging that can align. And therefore, they are going to be able to align and join. Now, a few things just to give you extra detail that the mark scheme's more particular on now is you need to know the enzyme ligase is going to be joining the plasmid and the gene of interest together. But specifically what ligase does then is it's catalyzing the condensation reaction, forming the phosphodiester bonds between those nucleotides to join those two fragments together. So now we've got our gene of interest into the vector, the plasmid, we have to get that vector into the host cell, which is the cell that's going to make the protein, which in this case is a bacteria. And in order to do this, we need to make the host cell's cell membrane more permeable, so we're more likely to get the plasmid in. And the way that we make the cell membrane more permeable to increase the likelihood of the plasmid getting inside is using calcium ions and heat shock, which means sudden increase and sudden changes in temperature. So that's going to affect the fluidity of the cell membrane, so we should be more likely to get the plasmid in. So we've now cloned it, but what we have to do is make sure that before we grow that bacteria, we are only growing the bacteria that has definitely taken up the recombinant plasmid. And that's what we mean by identifying transformed cells. A transformed cell is a cell that has taken up the recombinant plasmid. Recombinant means recombined. It's made up of two different types of DNA. Now, not all of them will be recombined or transformed cells because, first of all, maybe that recombinant plasmid, even though we heat shocked the cell membrane, it still didn't get inside. Or maybe a plasmid did get inside, but it wasn't a recombinant one. The original plasmid that was cut open simply rejoined, so maybe that got inside, so we don't have the gene of interest. And sometimes that DNA fragment that we created actually connects back around to itself and makes its own little loop of DNA. And if that went in, then again, it wouldn't work. So we have to identify which bacteria have successfully taken up the recombinant plasmid. And that's done using something called marker genes. And marker genes are on the plasmid and used to identify which bacteria have taken up the plasmid. There's 3 marker genes that are typically used. Genes that are coding for antibiotic resistance, genes coding for the creation of proteins that fluoresce, so they go bright yellow or green, particularly under UV light, and genes coding for enzymes that can catalyze reactions causing colour changes. Now, you don't need to know the details of how that is done, but it's a common application question. So that's why I'm going through this. You aren't expected to remember this, but it comes up as application a lot. So that's why I'm going to go through this example with you. So I'm going to do it with antibiotic resistance. And we can see that here is the plasmid. And there are 2 genes for antibiotic resistance. This one here makes the bacteria resistant to the antibiotic tetracycline. And this gene here will code for a protein that makes the bacteria resistant to the antibiotic ampicillin. And here is the DNA fragment that we've just isolated. Now, what they would do is deliberately insert that DNA fragment into one of the marker genes. And in this case, they've put it in the middle of the tetracycline gene. And what that does is our DNA fragment can be transcribed and make a protein, but the tetracycline one has been split in half now, so that is not going to be able to be transcribed and translated to make the protein that makes it resistant to tetracycline. So any bacteria that takes up this plasmid will not be resistant to tetracycline anymore, but the resistance to ampicillin gene is still intact, so it will be resistant to ampicillin. So to work out which bacteria have taken up the plasmid, the recombinant plasmid, you would grow your bacteria on agar, and we can see all the colonies here that have grown, and we've labelled them. You then use a sterile velvet block to make an imprint of this master plate and copy all of those colonies onto a second agar plate. And this time the agar plate has got agar with ampicillin antibiotic dissolved within it. And we can see that now some of the colonies have died and only some can grow. They would then take another imprint and this time grow those colonies on the agar plate that's got tetracycline dissolved within it. And now we can see only these 3 colonies can survive. And from this, we can work out which colonies contain the recombinant plasmid, because our bacteria that have the recombinant plasmid are resistant to ampicillin, because that gene's intact. So that means it has to grow on this plate, but it's not resistant to tetracycline anymore, which means tetracycline will kill it. So that means it won't be present on this plate. And that is only colony E and G, because they're on this one, they're not present on this one. So that tells us that E and G are the only colonies of bacteria that have that recombinant plasmid. So those are the ones that you should then remove and grow en masse. Next then is how you could do this, but using PCR. So PCR is our other way to get large quantities of the DNA fragment of interest. And vitro means not inside of a living cell. It's in a lab. So you can use the polymerase chain reaction to do this. It's an automated machine, so you get large quantities very rapidly. and PCR is going to be used probably multiple times per day in all genetics labs because it's so quick and you always want large numbers of copies of everything so you can do multiple repeats, and in case anything goes wrong, you've got your extra copies. So your PCR machine involves this thermocycler machine, and you would need to add your DNA fragment that you want to be amplified. You need to add the enzyme DNA polymerase, a particular type called Taq polymerase, primers, and DNA nucleotides. So Taq polymerase, this is a version of DNA polymerase that is taken from bacteria that are extremophiles, meaning they can survive in very, very high temperatures like hot springs. And as a result, they've evolved to have a version of DNA polymerase, Taq polymerase, that has an optimum temperature far higher. So we use this so that we can increase the temperature very high in this machine so the reaction happens even faster, but the enzyme doesn't denature. Primers, these are short sequences of DNA bases that are single-stranded, and they are needed to enable DNA polymerase to attach. So that's what we need to add into it. And then the process is, initially the temperature is increased to 95 degrees C. Now this is done so that the DNA fragment that you've added is going to have the hydrogen bonds break and it separates into two single strands. Then we decrease the temperature to 55 degrees C. And we need to do that so that hydrogen bonds can form, because our DNA primer, which is that short sequence of DNA bases complementary to our sequence on our DNA fragment of interest, those are going to be attracted and aligned, but we need the temperature to be dropped so that hydrogen bonds can form between those two. Then we will increase the temperature so that the enzyme DNA polymerase can then align all of these DNA nucleotides that are going to be attracted to their complementary bases. The DNA polymerase is then going to catalyze the condensation reaction, forming all of those phosphodiester bonds between the adjacent nucleotides. And that is at 72 degrees C. You do need to know those temperatures as well as this process. And that process will then happen on repeat. So you can end up with a huge quantity of DNA very rapidly. And this goes back to that maths formula that you can use. You would do 2 to the power of n, 2 meaning every time it doubles. N is the number of rounds of division that you allowed. So you could work out how many DNA molecules you'd have at the end. And if you didn't start with one DNA molecule, however many you started with, you would multiply by. So if we started with 100 fragments, we would then do 100 times 2 to the power of n. And if you left that going, let's say for 10 rounds, we would then do 2 to the power of 10 times 100, and that would tell you how many DNA fragments you'd have at the end. So the advantages are, it is automated, so it's more efficient, it's rapid, you can make 100 billion copies within hours, and it doesn't require living cells. That means it's quicker and less complex techniques are needed. So a few key terms as well that you need to be aware of. DNA probes, so this came up in the exams this year. They needed to define what a DNA probe is, and a lot of students struggled. A DNA probe is a short, single-stranded piece of DNA that is complementary to a specific allele or gene. Those probes are also labelled, usually with radioactive isotope or fluorescent marker, to allow detection. The process of DNA hybridization, this occurs when a DNA probe is going to bind to a complementary sequence of bases on a DNA sample. And to prepare the sample, the DNA is first extracted and heated so that it is separate strands. Then the probe is added so that it can bind and hybridize. And the point of doing this is you can then use the presence of that probe on a sequence of DNA to identify a target allele. And that's because if you've labeled it with a radioactive isotope, when you use X-ray film, it will be visualized, or if it's a fluorescent marker, it will visualize in UV light. So it's used, these DNA probes are used in medical genetics to screen individuals to see if they have particular alleles linked to inherited conditions such as cystic fibrosis or Huntington's disease, or the gene here linked to breast cancer. They are also used to identify variations in genes that affect drug response, allowing doctors to predict how a patient might react to certain medicines. And this kind of screening can help to detect individuals at increased risk of disease, even before the symptoms occur, which allows for early intervention, monitoring, or even preventative treatment. So this is what we mean by personalised medicine and genetic counselling. We're screening for the presence of that particular allele, selecting personalised medicines based on your genetics. And that's because some drugs are more or less effective depending on your genetics. And that enables it to be more effective to you, but also more cost effective as a treatment. And genetic counselling is a type of social work giving people advice and information following their screening of disease-causing alleles. So then we go on to how this process might occur using gel electrophoresis. And VNTRs are needed for this. VNTRs are variable number tandem repeats, and an organism's genome contains many regions of non-coding DNA, which are VNTRs. So they're short sequences of DNA bases repeated a variable number of times among individuals. And the number and length of the VNTRs is very different from person to person. And that is very useful in DNA analysis because VNTR patterns are inherited and highly specific. So the probability of two unrelated individuals having the same VNTRs is extremely low. So we can use VNTRs to examine relationships in terms of genetic similarities. Genetic fingerprinting is the technique used to analyse the DNA fragments containing VNTRs. And the process starts with the amplification of DNA using PCR so that you've got a large sample of DNA. The DNA is then cut into fragments using restriction enzymes, separating the DNA at specific sequences. And those fragments are then separated using gel electrophoresis, which will separate them according to their size, so the number of bases in the VNTRs. A DNA probe is then added, which binds to specific VNTRs, and the pattern of the bands forms a genetic fingerprint. And this pattern is unique to an individual, except for identical twins. So let's go through that whole process in more detail. Genetic fingerprinting, so first of all, you would collect your DNA sample. And the smallest sample of DNA can be collected, and that's because we're going to use PCR to amplify it afterwards. So any cell that contains a nucleus can be used. Then we have the digestion stage once we've collected and extracted the DNA. And the digestion stage is when we use restriction endonucleases to cut that DNA up at the recognition sequence or restriction sites. And enzymes which cut close to your target VNTRs will be added, and we get that palindromic sequence with those sticky ends. Now we need to separate out all of those cut up VNTRs. And the DNA samples are loaded into these tiny wells in agar gel. The gel is then placed in a dish which contains this buffered liquid, and an electrical voltage is applied. The DNA, because of that negative charge on the phosphate group, is going to be attracted to the positive end, and therefore the DNA is going to move towards this positive end through the gel. And the reason that it moves is, first of all, DNA is negative and it's attracted to the positive end, but it moves different distances because of how many DNA bases there are. The bigger a DNA base sequence, the heavier it is, so it's not going to move as fast or as far. If it's a shorter piece of DNA, fewer DNA bases, and that means it's lighter, so it can move through the gel much faster and it'll get further along. So that stage is gel electrophoresis, and that agar gel creates the resistance, and this is what I was saying about the smaller pieces can move faster and further. And that is how different lengths of DNA or VNTRs are separated. We then add an alkaline to make all of that DNA single-stranded. Then we can add our DNA probes to align to those single strands. So we said the DNA probes, those short single-stranded pieces of DNA, complementary in base sequence to the VNTRs. Those probes will be radioactively or fluorescently labelled. So then if they hybridize, we then rinse it afterwards, and then the agar gel, this bit you often don't get assessed on, but the agar gel will eventually dry out and crack. So we do actually transfer it usually to another material that is longer lasting, and that can then be used to visualize your results. And here's one that I did over 10 years ago now, which is absolutely mad. I can't believe that was 10 years ago, but this is looking at the different VNTRs in bacteria, and we could then analyze it to see, here are our knowns, here's my unknown, and I could see my unknown had the same DNA bands as bacteria number 3. So I knew my unknown bacterial sample was the same species as bacteria number 3. So that can be used to interpret paternity tests as well. So we could look at the mother, the child, and any band that the child has that isn't from the mother must be from the father. And that's how we can use it to work out which person must be the father. And I think actually in this one, there aren't any that are a perfect match. Number 1 almost is, but there's this band here that isn't in number 1. So the uses of this genetic fingerprinting could be paternity testing. It could also be forensic science to identify if someone has their DNA at a crime scene, to place them at the crime scene. Medical diagnosis to see if you have particular alleles linked to inherited diseases to help with early diagnosis. Animal and plant breeding to make sure that you aren't about to breed 2 individuals that are very closely related. And also assessing genetic diversity to look at how much genetic diversity there is in a population. So that takes us to the end of topic 8. Hopefully you found it helpful, and if you did, don't forget to scan here for my mark scheme specific notes, which also have recall questions, examiner tips, summaries, key marking points, and essay links. On my flashcards, scan here, where you can get mark scheme specific key points. But that is it for today's video. Hope you found it helpful, and I'll see you in a video very soon.